sexual reproduction in plants, if we see this topic, we have to understand that this, at the end of the day, what are we having? We have the flowers. Inside the flowers, we have the male and the female, and then fertilization happens and it gives us the seed and the fruit, right? Why do we grow a plant? We have some benefit out of it, right? We consume the leaf, we eat the fruit, seed, of course, right? Yes. So students, this sexual reproduction in flowering plants, the first ever question they asked you is, are you really happy? The main reason I asked that is because these flowers not only is just enjoy the beauty of the flowers, but understand they help us culturally. That means in many of our Hindu cultures, even Christian cultures, even in Muslim cultures, be it any culture, they use flowers to indicate something, right? Even if it is for love, even if it is for affection, even if it is for grief, we use these flowers. Yes or no? All of you agree to it, right? At the same time, they're aesthetically beautiful. Okay, the students, this and all is not, see, NEET questions, but this is the starting of the chapter. Okay, they are ornamental. They are ornamental. Okay, they are used for a culture, aesthetic ornaments. They are also used to express different kinds of emotions, right? Different kinds of emotions. How many of you have got a flower from your parents, loved ones, from your friends, and you felt very excited? Very excited. Mike, voice is breaking. Now it's fine, huh? Maybe because I put this inside. 1 second, let me check. Okay, tell me now. Is it fine? I think it's because the mic is moving. But if I don't think, if I put, student, 1 second, I'm checking, okay? You felt very excited. Very excited. Mike, voice is breaking. 1 second, 1 second. I'm checking, I'm checking. I think it should be fine now. I think it's because the mic is moving. Yeah, whenever I turn, no, there might be a slight issue. Otherwise, it's all fine. Okay. Yes. So students, to to show different kinds of emotion, be it love, be it happiness, be it sadness, even for people's death, right? We go with your bouquets. Even for wedding we use it. So flowers are basically to express any kind of emotions. I asked all of you a question: have you ever received a bouquet from someone like your parents or someone you loved, or someone like who is so dear to you? You will know that emotion. So this chapter starting is talking about all of that, which is again not very neat point of view, but you should just know this introduction to this chapter. Are you all with me on this? Yes, awesome. Now, sexual reproduction means what, students? Sexual reproduction means, simply means, be it in animals or in plants, it basically has only one meaning. That is the male gamete, right? The male gamete, and there is a female gamete, and this both are fusing and giving us a zygote, right? A diploid zygote, because this will be N, this will be N, and this will give us 2N. This is the ultimate, or this is the simplest definition of sexual reproduction, right? Sexual reproduction. Yes. Okay. So male, female fuse together, male gamete, female gametes fuse together, they do fertilization, and at the end of the fertilization we get a zygote, and then continues. This is your sexual reproduction. So this was the introduction to the chapter, nothing much. Now students, we are going to start the chapter on the note of flower. Now flower, mainly if you see a plant, generally if you see a plant, it has two parts, okay, two parts. Okay, full, all of them are like, oh, thank you students, thank you for sending me flowers, so beautiful of you all. Okay, very nice. Now all the people who put flowers, now put attendance in the class. Okay, attendance in the class. Yes or no? Yeah. You know, with that plants, that part also stem, some people take the stem to hit. Remember, your parents used to take that, right? They used to go outside, they used to break something, and they used to come back, right? A branch of a plant, a branch of a plant, you would have got hittings also. So don't only think about the good memories, think about the other beautiful ones also, getting hittings from your parents. Now students, flowers, or say a plant, no, I would say a flower, forget about plant, flower only we'll say, okay? Has two parts. Essential and non-essential. Essential and non-essential means what? Essential means it is directly involving in the reproduction of the plant. Non-essential means it's not directly involving, but it is indirectly involving. Okay, because many students, you know, whenever they see the word non-essential, they're like, useless. Why will I study about it? No, not at all. Essential means they're directly involved in the reproduction, but non-essential means they're directly not involved in the reproduction, but they're helping in reproduction. Now, let's see what is this essential and non-essential. Students, essential is your androecium. Okay, androecium and gynoecium. Okay, gynoecium. Essential is your androecium and your gynoecium. Is this clear? Yes, awesome. Okay, now when I see non-essential, when I say non-essential, whom do I talk about? I talk about calyx and corolla. So simply means that essential part is directly involving in reproduction, but non-essential part is not directly involving reproduction, but they are actually helping. Now let's see how they are going to help. Okay, how they are going to help. Student, look here, androecium, gynoecium. Androecium is the male part of the flower and gynoecium will have the female part of the flower. Here if you see non-essential, you have calyx and corolla. Now calyx, who forms the unit of the calyx? The unit, all of you would have seen this, right? Sepals. Sepals constitute, see, these are the whorls of a flower. Whorls of a flower, you have androecium, gynoecium, calyx, corolla. They have multiple units as well, right? Now this is a class. Okay, this is a class. So many students are in this class. Gopika ma'am's class is there. This is the class. Inside a lot of students are there. You people are becomes the unit of the class, right? So same way, calyx is the whorl and it is made up of a unit of sepals. Okay. Now if I draw sepals, all of you would have seen this. Yes, all of you would have seen this in a plant. This is your sepal. Okay, then if I say corolla, who doesn't know corolla? Corolla forms the petals, right? Corolla, the whorl corolla will have petals as its unit. Now you people tell me, don't you think these two people are actually helping in the entire reproduction process? Because how do an insect get attracted to a plant? An insect get attracted to the plant by looking at the flowers. How do you people know that it is rose or, you know, sunflower or all that? Looking at the flowers, you find it very beautiful, right? Earlier you people used to take photo, no, like this, used to hold hibiscus and stand in Facebook and tell being loved with 10 other people. Okay, I used to always remember seeing, I think our parents, okay, mainly our parents, we are all little aesthetic people, but if you see our parents before Facebook post, they will have one hibiscus plant, they'll be holding on that hibiscus plant and they'll be like feeling loud with 10 others. They don't even know who that 10 people are. Whoever they found, they put. So flower is an indication that we have gone to some place. Now if we go to Kodaikanal, Ooty, other places, anywhere they see a tree or a flower, immediately our parents will be like, you take photo near, you take photo here, okay, near the plant, okay, and then I'll put. So exactly. So that was, now we find it very, we tell cringe, weird and all the words we use, fancy words, but back then we all have done it. We all have done it at least once in our life, holding a flower, standing near a tree. So flowers are a beautiful indications, okay. So yes, corolla is your petals, the units are petals, okay. Now when I talk about androecium and gynoecium, students, androecium we have stamens, okay, androecium the units are stamens, and in your gynoecium it is your pistil or carpel. Pistil or carpel, okay, pistil or carpel, yes or no? True. Okay, so those who joined right now, quickly smash the like button. Yes, kind of. Okay, someone is like, oh, ma'am, I didn't do it fully. Like, kind of, I have done it in my life. Okay, boys editing is next level, students. Girls used to stand near flower, keep flower on the head and take photo. Boys used to, what they used to do, they used to put that Batman outfit. Then they used to stand on a bike, not even sit on a bike, stand on a bike, hold one whole tree on their hand like this. So the editing skills in before COVID used to be like a meme by itself. Okay, so yes, so these are the entire constitution of it. Now if I draw a flower, students have to draw a flower. Okay, so I'm going to draw a flower. I'm not going to draw your epigynous, superior ovary, inferior ovary and all. I'm just drawing this for the labeling part, okay? That much botany also we don't need in life. Yes. And now let's draw the boy. Anthers. Okay, anthers are there. Okay, anthers are there. Yes. Now let's draw the petals. Only 2 petals, okay? Special flower this is. Yes, this is a flower. Yes, okay. Okay, yes. Fine. So students, let's label the parts. Okay, let's label the parts. See, now I've drawn the receptacle and thalamus. Now it is open, like it's broad enough that I've drawn, but you know it is not this broad also. Okay, this is just to make you label easily. Okay, so 1 second. If I this is your receptacle or thalamus, which will be a swollen part of the flower, right? Swollen part of the flower. Yes. Yes, done. Okay, let's label each of the parts. So this is the receptacle or the thalamus, which is a swollen part. Okay, which is a swollen part, and from there, receptacle or the thalamus. Okay, thalamus. From there, if you go to the top, you have this bulged portion or the swollen portion, which is the ovary. Okay, which is the ovary. Now students, inside the ovary, the ovules are going to be there. Okay, inside the ovaries, who is going to be there? Ovules will be there, and they can have different kind of arrangement. They can have different kind of arrangement. Now inside this, ovules are there. Ovules are there. This long slender structure that you're seeing here on the anther is called as your filament. I know this you would have studied in your 6th standard EVS and all that, but you have to study it now also, okay? And then this portion is your style. Girls never leave style, yeah. Style. Stigma. Stigma. Okay? This is anther. This is your anther. Yes or no? Yeah? Stigma, style, ovary, your anther, filament, your receptacle, thalamus. This is the parts of the flower. Are we done? Okay, are we done? And then you have inside the ovary, ovules. Clear? Easy, no? These are all simple things that you have studied before. Yes, okay. Now students, one more word you have to know. If I show the filament, right? This is the filament and the anther. This becomes, this portion which is attaching, the filament end which is attaching to the thalamus will becomes the proximal end, and the one that is far from it, right? Far from the attachment will be called the distal end, okay? Something that is at very, distant, right? Very distant. That will be called as the distal end. The one which is closer or attached to something will be called the proximal end. So if someone asks you, if the question comes about the proximal end and the distal end, you have to know this part becomes the proximal end. That means where the filament is attaching to the receptacle or the petal or something, that becomes the proximal end, and the one that is far away is called the distal end. Yes or no? So I'll draw it down and I'll show you. So this is your filament, this is your anther. Okay, this is your filament and this is your anther. So here, say this is your receptacle. Okay, I'm just drawing it like this. Okay. Now this becomes the portion which is attached. This becomes the proximal end. Actually, questions have come on this. Okay, don't be like, hey, what is ma'am telling? This becomes the proximal end and the anther becomes the distal end. Okay, distal end. Now, students, when the plant is grown itself, they know, even if the flower is not arising, they already know that the flower has to be formed. That means the fate is already decided. The moment the fate is decided, they will immediately start doing the hormonal and the structural changes in that plants. Okay, now you have already decided to crack NEET, you have already, adjusted yourself to do that, right? The fate, you becoming a doctor is already decided on that you're preparing. Same way, even at the plant, the moment the plant is grown, we know, or the plant decides that flower has to be formed, immediately the hormonal and the structural changes will be done for it. Okay, awesome. That is the basic thing about it. So students, this is your flower and this is the main non-essential and the essential part of the flower. Now, all the flowering plants, one statement is given in your NCERT. How many of you remember this? Okay. All the flowering plants are sexually reproducing. Yes, flower is a sexual or reproductive organ of a flower. Reproductive organ of a flower. Is this clear? Okay, is this clear? Now these are the units, okay, these are the units. Now your pistil, your stamen, pistil again has multiple parts, okay, that we are going to do again here. Now students, there are pre-fertilization events. So understand, in this chapter you will be studying 3 events, okay, 3 events. What are the 3 events? The events goes like this. First event is going to be a pre-fertilization event. Fertilization event. Simple, like before the marriage, right, we do a preparation. Before the exam, we do a kind of preparation. That becomes a preparation, right? So, pre-fertilization event. Then your second event is the fertilization. That means actual time when you're writing your exam, when the marriage has happened, okay, fertilization. And the third becomes the post-fertilization. After your exam, how things are going, like you'll be relaxing, okay, or you'll be waiting for Supreme Court hearing. So, depending on what situation our life is, these things will happen. Okay. So, 3 stages are there: pre-fertilization, post-fertilization and pre-fertilization, fertilization, post-fertilization. Pre-fertilization means, as I told you, the plant has already decided to form a flower. Immediately, they'll start making the structural and the hormonal changes. Okay, clear? Yes, very good. Pre-fertilization, fertilization and post-fertilization. Awesome, awesome. Okay, awesome. Yes. Yeah, the likes are very disappointing. Yes, like now, exactly, Hasini, that's why I told this, okay. Okay, so this is already decided by the plant that, okay, the flower has to be formed and we will do that, okay, we will do that. So here in the pre-fertilization event, we would say that if the flower has to be formed, it will be ready to form the floral primordium. Okay, floral primordium. Primordium means an organ or a tissue. So they are already setting up the whole process. They are setting up the whole process. Now you should understand, students, in your floral primordium, there is something called as inflorescence. Okay, if I put it in bracket, you will think same word it is. Okay, there is a word called as inflorescence. All of you know this word inflorescence, yes or no? Inflorescence. That means it's very simple. See, what is the goal of the flower, or what is the role of this flower? To attract insects, to bear the male and the female organ. But there are flowers where the flowers are very, very small. Very, very small. Now the insects will not be able to see it, or the insects will not be able to attract, okay, get attracted to this. So that what these people will do, they will do a bunch of inflorescence. That means a bunch of flowers which are arranged together, and they will look beautiful, they will look attractive, they will look large enough to attract an insect. So inflorescence are of 2 types. Which are the 2 types of inflorescence? Rani, I have sent the Telegram link, no? I sent you the Telegram link and asked you people to join. Are you not on our Vedantu NEET English Telegram and Biology by Gopika? Students, join the Telegram so that you do not miss any kinds of update, and you will get all these notes only there. Okay? Nowhere else you will get these notes. Got it? So students, inflorescence are of 2 types: racemose inflorescence and cymose inflorescence. What is racemose and cymose inflorescence? All of you tell me in the chat, what is racemose and cymose inflorescence? Quickly, I'm waiting for your answer. Yes, I have sent the Telegram link. I had sent the message on the Telegram. Yes, answer, all of you. Who is racemose? Yes, very good. Okay, okay. Yes, many are telling different answers. Exactly. In racemose, what happened? The axis is growing continuously. Axis is growing continuously, right? And what is happening? The younger flowers will be on the top and the older flowers will come down. They'll be like, okay, my time is over. So if this is the axis, students, here the younger flowers will be there. Okay, they will keep growing. Indefinite growth will happen. Okay, here the younger flowers will be there. And the older flowers will be to the down. Okay, older flowers will be downwards. They'll be like, okay, we are old, you people are young, so you people keep growing. So this will be an indefinite growth, okay? It will keep continuing to grow. But in the cymose, it's not like that. Cymose, what's happening? The older people will be towards the top. That means the older flowers will be towards the top and the younger flowers will be to the bottom. Okay? The younger flowers will be to the bottom. This is called as cymose. If you notice nowadays on on the all social medias, online medias, you will see some parents dancing, like some grandparents will be dancing, parents will be dancing. They have so much energy at this age also, 40, 65, even at 70 people are dancing. So this cymose is like that. The older people will be like, you shut up and be down, I will go up. I am very smart and active, you do not call me old. So here what is happening? Older flowers are to the top, younger flowers are to the down. Okay? Yes? Awesome, awesome. Okay, so students, this is the type of inflorescence. This inflorescence is basically for only one purpose, to attract more insects, because the flowers are too small and insects will not get attracted. So they form a bunch to continue this process. Okay, now when I was talking about pre-fertilization event, as I told you, flower has to be made. That's hormonal and structural changes will be done. Hormonal and structural changes will be done by the plant. Okay, this class also has a speciality. The speciality is that I have put NCERT SNP. So we will be studying NCERT as well as I will be teaching and drawing and teaching you. So this way you will not have to go back and read your NCERT. Okay, done. This is the main task of today, where I will be teaching you NCERT. At the same time, I will also explain and teach you. Got it? Yes, very good. Very good. Okay, so here students, androecium, gynoecium, all this is getting to form because that is a pre-fertilization event. Fertilization is going to be the next stage, so we have to do something before that. It is the formation of your androecium, gynoecium, flowers, all the structural hormonal changes, all this. Done? All the structural hormonal changes and all of it. Okay, now I am writing it here. Is blue visible? Okay, so formation of androecium and gynoecium. Androecium and gynoecium. So pre-fertilization event, there is a paragraph given in NCERT. Let's just see the paragraph. Much before the actual flower is seen on the plant, the decision, okay, the decision that the plant is going to flower has been taken place. Plants are more organized than us. See, plants know what to do, when to do. Several hormonal and structural changes are initiated which lead to differentiation and further development of the floral primordium. Okay, floral primordium. Primordium means any organ or a tissue. Okay, floral part. Understood? Inflorescence are formed which bear the floral buds and then the flowers. So all this decision have been made. Okay, and also any kind of modification that has to be made also will be made by the plant. Okay, in the flower, the male and the female reproductive structures, that is the androecium and the gynoecium, differentiate and develop. You would recollect that the androecium consists of a whorl of stamen representing the male reproductive organ and the gynoecium represents the female reproductive organ. So understand, these are whorls. Androecium, gynoecium is a whorl, and inside that there are separate units. Okay, inside that there are separate units. Now let's talk about stamen. What do we know about stamen? So students, stamen basically has two portions, right? Two parts. What is it? Anther and filament. Anther and filament. Now if I talk about anther, I have to talk a lot about anther. But first we will draw the structure, right? Let's draw the structure. This is my filament. Okay, and this is going to be the anther. If you look at the anther, don't you see that there are two lobes in the anther? Yes or no? The diagram I drew, it has two lobes. Okay, it has two lobes. Two lobes are definitely present. So can I call it bilobed? Can I call it bilobed? Yes, ma'am, you can call it bilobed because anther has two lobes and I will call it bilobed. Okay, now if I take a cross section of this anther, if I take a cross section of the anther, or if I cut it longitudinally, I will be able to see thekas in it. So how do I see it? Now if I take a cross section of it like this, okay, cross section of it like this, and if I draw it, okay, if I draw it, I will be able to see something like this. Okay, this is the filament again, and this will be the anther. Students, I'll not be able to draw exactly like that. Okay, something like this I'll be able to see a cross section, right? A cross section I had taken. The top part also is here. Okay, top part is also is here, and here also there is the top part. Yes, this will be the cross section. Now when I did the cross section also, I'm able to see some different types of lobes or thekas. So that is, in each of this there is going to be a theka. There is going to be theka. Yes or no? So 4 thekas are there, that means 2 in each lobe. Any doubt in this? Any doubt in this? All of you tell me in the chats. Very good. No doubts, ma'am. Okay, no doubts. So it's a bilobed structure. Dithekas they are telling. Okay, that means in each lobe there will be 2 thekas. So let's say I erase this diagram and I draw a completely, complete structure of how the anther would look like. Something like this it would look like, students. Okay, they would look like this. Okay, they would look like this. And if I draw the 2 lobes and if I draw the thekas, they would look like this. Okay, they would look like this. See? And those 2 lobes are connected with the help of a connective. Okay, there's a connective in between which connects them. So, what and all do I know about anther? I understood that the anther is bilobed because 2 lobes are there, they are bilobed. Okay, they are bilobed. After that, I got to know that each lobe has 2 thekas. So, I am going to call it dithekas. I am going to call it dithekas. And if I call, students, these each thekas can also be called as sporangia, okay, sporangia. So we call it tetrasporangiate, okay, tetrasporangiate. Now microspore, all that we will come into, but right now understand this anther is also tetrasporangiate, tetrasporangiate. Yes, clear? So, the anther is bilobed, dithekas, tetrasporangiate, and we can also use the word tetragonal. Tetragonal. Is this clear? Anyone has any doubts? Anyone has? Yes, very good. Tetrasporangiate. Okay. Now, students, each of this. Now, ma'am, what is this theka? Why are we using it? What's the purpose of this? We need to know the purpose of this, right? The purpose of this is, students, this theka or your microsporangia, okay, microsporangia later develops to form your pollen sac. So these are the people who are going to store your male gametes later once they mature. Okay, so when I'm labeling it, what am I going to label it as? I will label it as microsporangia, okay, later forming pollen sac during maturation. Okay, pollen sac. But before this also you should understand, when the anther is young, okay, when the anther is young, this is a very important line. When the anther is young, some compactly packed, okay, some compactly packed cells will be there. Compactly packed cells will be there. Students, that cells are called as sporogenous tissues. Many cells will be there, okay, sporogenous tissues. This is when the anther is young. Okay, when the anther is young, they will have some compactly packed cells. Okay, compactly packed cells, and these cells are called as sporogenous tissue. This sporogenous tissue later during maturation forms your microspore mother cell, again undergoes meiosis, then form your microspore. This microspore ends up forming your pollen grain. So this is all about microsporogenesis, okay. But if you don't understand this, then you will not understand this. Okay, you will not understand the rest of it. Is this clear? Please look at the board and let me know if it's clear. Everyone in the class, is it clear? Yes, all of you. Okay, okay. This will be your microsporangium, which later during maturation will become pollen sac. Okay, pollen sac. And then we have, when the anther is young, there will be a set of cells which have accumulated, compactly packed cells, which is called as the sporogenous tissue. Okay, this is all about it. Now, if I have to talk about filament, how can I ignore the filament? Okay, this filament, students, is basically like a stalk kind of a structure which will hold your anther in place. Okay, this filament is long, slender, and provides the support for the anther. Okay, it provides the support for the anther. Got it? That's it about your filament. Okay, clear? Any doubts? Yes. In the filament, only one thing you have to understand: proximal end, distal end. This one question had come, so you should understand this is the proximal end, this is the distal end. Filament is long and slendered. Okay, the stamens combine, many units of stamens will combine. These are the units. Okay, they will combine to form your whorl called as androecium. Okay, androecium. Okay, now that we understood about the anther structure, let's see what NCERT is telling. Students, after this class, you will not have to read NCERT. I assure you that you don't have to read NCERT. See, a typical angiosperm anther is bilobed. Yes, all of you understood why it is bilobed, okay? Which each lobe having two thecas. Yes, ma'am, this also I understood. That is, they are dithecous. Don't tell tetrathecous, okay? They're not calculating for the whole. They're calculating each lobe has two thecas, so we call it dithecous, okay? Often a longitudinal groove runs lengthwise separating the thecas. Let us understand the various types of tissues and their organization in the transverse section of an anther. They're telling, usually if this is the anther, okay, this is the anther, there will be a longitudinal line that runs like this, okay, like this. But now we are going to study this, okay, we are going to study the transverse section. Clear? Okay, transverse section of the anther. The anther is four-sided. The anther is four-sided, so we will call it tetragonal structure consisting of four microsporangia. Four microsporangia located at the corners. Okay, corners, two in each lobe. Agreed? So far good. Two in each lobe. Yes? Yes. Okay, fine. Now, the microsporangia develop further and become the pollen sac. So, only during the mature stage, I can call it pollen sac. Otherwise, I am supposed to call it microsporangia. Okay, they extend longitudinally all through the length of the anther and are packed with pollen grains. Okay, are packed with pollen grains. So far good. Okay, so if I take a cross section like this, students, I'll be able to see a structures like this. Okay, I'll be able to see a structure like this. I've separated it. Inside this, what will I be able to see? I'll be able to see a lot of pollen grains. Okay, during maturity, I'll be able to see a lot of pollen grains. If you people have seen hibiscus and if you have touched the anther, you would be able to see that white powdery structure on your hand. That is your pollen grains. Done? Done and dusted? Okay, chalo, let's go to the next one. Structure of a typical anther. Now, I have already done your typical anther, but we are going to understand more about microsporangium. Okay, we are going to understand more about microsporangium. Is this clear? So, we will learn the layers as well as the structure. So, students, this is the structure. Let's draw it once again. Okay, let's draw it once again. A big structure we are going to draw. Okay. Yes. We've drawn the structure. Can you see? It's not exactly like butterfly shape, but they have one side up and down. Here we have the thecas or your microsporangia and the corners. Okay, you should always draw it in the corner. Okay, now we will just label this. Okay, these are microsporangias. Sporangia. When the anther is young, okay, when the anther reaches maturity, okay, when the anther reaches maturity, I will be able to call it pollen sac, which are filled with pollen grains, okay, which will be filled with pollen grains, okay. Now, how is this both lobes connected to each other? They have a connective, right? Each of the lobes are connected with each other with a connective in between. Connective in between. Is this clear? Any doubts? Ask me now. Okay, this is the connected. Connected. Now, this so far good? Yes, layers we are going to come into, okay? Layers we are going to come into, okay? Okay, thank you, GT. Now, students, look here. Microsporangia, pollen grains, understood? Okay, this is done. 4. What and all do we know about anther? Let's write that also. What and all do we know about anther? We know that the anther is bilobed, okay? Bilobed, tetrasporangiate, tetrasporangiate, tetrasporangiate. It is tetragonal, okay? Tetragonal. Tetragonal, and one more word was there. Who can tell me the one more word? Yes, all of you. 4 layers. Yes, we are going to the layers. Before that, tell me one more word. Bilobe done, tetrasporangiate done, tetragonal done. One very important word is that. Starts with D. All of you. Very good, Madhumita. Diathecus. Diathecus. Yes, I do not see Madhumita's comment. I do not see Hasini's. Yeah, Hasini's comment I saw. Pavitra's comment. Okay, you people are not commenting. Jay's comment. You people are not commenting well on my videos. I do not see your names. Okay, Neha, Nayana. Where is all of you people's comment? Okay, so students, anthers are bilobe, tetrasporangiate, tetragonal and diathecus in nature. This is all about your anther. Now we are going to learn the layers. Okay, now we are going to learn the layers here. What are the layers of this anther? Students, this anther, if I again draw a small structure here, okay, like how many times I drew heart, now I have to draw this. Okay, this is an anther, and if I take this portion, okay, this portion of the anther like this, and I do an enlarged structure of this, I know that this anther is made up of around 4 layers, okay, protection, 4 layers are protecting it, around 4 layers are there. And who are the 4 layers? The 4 layers are epidermis, okay, endothelium, middle layer, and tapetum. These are the 4 layers of your anther. These are the 4 layers of your anther. Yes? Oh, awesome, Hasini. Elephant eats my tomato. I had told this in last class, okay? When before, when I was teaching for 2024, I had told elephant eats my tomato because many students get confused between epidermis, endothelium and middle layer. So I thought, okay, I'll make one way to remember for this. Okay? Students, in that, your epidermis is a single layer. Okay, epidermis is a single layer. Then we have endothelium. Okay, we have endothelium. Again, endothelium also is a single layer. Then we have middle layer. Okay, middle layer, which is around multiple layers. Okay, multiple layers are there. Say I draw it like this. This is your middle layer. One more layer I can draw. Middle layer can be 2 to 3 layers. Okay, middle layer, and then we have your tapetum. Okay, then we have our tapetum, which is a nourishment layer, which provides nourishment for the growing embryos, for the growing pollen grains. Okay, pollen grains. Now, students, what is so special about these 3 layers? We are going to write the ploidy of this level because I saw many questions on the ploidy. Now, what happens is we tell so many other things about this layer, we forget about the ploidy. So, now we'll write about the ploidy. But before that, I want you to understand epidermis, endothelium and the middle layer mainly does two functions. Okay, 2 functions. What is that function? They help in protection. Okay, they are mainly for protection and dehiscence. They mainly for protection and dehiscence. These 3 layers, epidermis, endothelium, middle layer, mainly for protection and helping the anther to dehiscent. But what about tapetum? Tapetum is not like these people, okay? Tapetum is little special because tapetum provides the nourishment Okay, tapetum provides the nourishment. Not only nourishment, students, it also releases some kinds of enzymes to help us in microsporogenesis. Okay, it helps us with nourishment of the pollen grain growing for the, they help the pollen grains to grow. They provide the nourishment. At the same time, okay, at the same time, they also help in releasing some enzymes, okay, and some enzymes for your microsporogenesis. When I teach you microsporogenesis, you will understand this. Awesome. Good to go? Yes, good to go. Very good. Yes, nourishment. Nourishment. Students, will draw one box and write it. Okay, we'll draw one box and write it. Are you all ready? Yes, nourishment, nutritional. So this is the layers of your anther. Okay, layers of your anther. Students, so sad. Anther looks so bad without microsporangia. Okay, microsporangia. Yes. Now, I'll show you the diagram in your NCERT. Look here. Your NCERT diagram. You can see that a small portion is taken. Okay, the first layer is your epidermis, endothelium, middle layer, microspore, mother cell, and your tapetum. That is, when they took a small portion, a microsporangia also was taken half, right? So, in the microsporangia, what will be there? Your microspore mother cell will be there. I told you the sporogenous tissues have compactly got arranged. Later, during maturation, they'll become your microspore mother cell. Okay, done? Done? Now, we'll draw one box. Okay, we'll draw one box. Okay, 1 second, students. Okay, here only we'll draw the box. Okay, I've put heading microsporogenesis, but here we'll draw a box to know about each of the layers. Okay, all the layers you have to know. Yes. 1, 2, 3. Yes. Okay, done. Now let's write. All of you, I want you to write this with me. I know you will get the notes, but still write it with me. We have to know few things about these layers. Okay, we have to know few things about these layers. These are the things we are going to know. Yes. Okay. What are the things we have to know? Okay, what are the things we have to know? So, students, first we will write layers. Okay, layers. Next we will write their function. Okay, function and more about it. And then we will write their ploidy. Okay, ploidy. Yes, ready? All of you? All of you ready? Yes, ma'am, ready. Okay, chalo, let's start about the first wall layers, that is the epidermis. Students, epidermis is like the principal. Okay, epidermis is like the principal. Okay, principal is single and it is the outermost layer and it has more authority in protection. Okay, so it is the outermost layer. Outermost layer. And it does mainly protection. It does mainly protection. Now, can anyone tell me the ploidy level of epidermis? Can anyone tell me the ploidy level of epidermis, all of you? Anyone try? Just give it a try. So, epidermis, it is the outermost layer and it is going to do the protection of the pollen sac. Okay, they are going to do the protection of the pollen sac. And students, this is a single layer. Okay, this is single layer. Even your endothelium is a single layer. Why you have to know single layer? Because you have to know which is multi-layered. We have to know which is binucleated, multinucleated. All this to know only. I am telling again and again, this fellow is single. Who epidermis is single layer. Students, the ploidy is diploid. Okay, diploid. Diploid is the ploidy. Clear? Any doubts? Yes, very good, very good. Kuvali, who was that? Diploid. Some people told haploid. Students, diploid. Two. Wow. Ma'am, haploid or diploid? You choose. Okay, because you are the teacher, you should be able to know this. You choose. Okay, I don't have time to choose. Fine. So, students, epidermis done. It is the outermost layer. It does the protection of the pollen sac and the ploidy is diploid. Okay. The next one is endothelium. Endothelium. This layer is the layer next to the epidermis. Yes or no? Okay. It is present inner to the epidermis. Now, what does this layer do? Okay. This layer is inner to the epidermis. Inner to the epidermis. Students, I am writing, I could have just taken the NCERT and read, but I am writing and making you read NCERT. So all you have to do is, after the session, let me know how was my botany class, okay? And you should also let me know if you understood everything, fine? Okay, so inner to the epidermis, students, and this fellow mainly does the job of dehiscence. Okay, what does it do? What does it do, all of you? Mainly does the job of dehiscence. Now understand, if the anther does not break open, then the pollen grains will be never released. So we have to make sure that that hygroscopic nature of anther, no? That means we have to dry out the anther so that the anther will break. So this people mainly helps in dehiscence. Dehiscence. This one's ploidy level is also diploid. This one's ploidy level is also diploid. Okay, done? Next layer is which one? Middle layer. Okay, middle layer. Middle layer is little more special. Okay, it is inner to epidermis, but the thing is, this layer is not a single layer. Yes, it is not a single layer. All of you are with me on this? Yes. Okay, ma'am, it's not a single layer. This layer, this has around 1 to 3 layers. Okay, students, it is 1 to 3 layers. Okay, first I'll write it's inner to epidermis. Wait, okay, it is inner to epidermis. This question had come, not this inner to epidermis, the next one I am going to tell. It has around 1 to 3 layers, and students, this is a short-lived layer. Okay, this is short-lived. So the question had come, which is the short-lived layer. Okay, in your anther, the answer should be middle layer, because it has around 1 to 3 layers, and this layer is short-lived. Okay, this layer is short-lived. Clear or not? Audible Rani, just leave the session and join back. Yes, 1 to 3 layers are then, and the cells of these layers are very, very short-lived. Okay, very, very short-lived. This person also helps in both protection and dehiscence. Okay, this also mainly helps in protection and dehiscence, but if you have to say a specific character, it degenerates in maturity. Okay, that is also one important point. It degenerates in maturity. So once this mature, this layer will get degenerated. This layer will get degenerated. All of you are with me on this, yes or no? Okay, this is also diploid in nature. Diploid in nature. Okay, now the last one is tapetum. Actually, tapetum is the hero. We have to write it separately, but I thought no need to do partiality with others. Tapetum. Yes, students, tapetum is a very special layer. First of all, understand, it is not going to do protection or dehiscence, but its main job is to do nourishment. Okay, and it is an inner layer to the epidermis. This layer does nourishment. Okay, nourishment, mainly nourishment. This is the main task of tapetum, to do nourishment for the growing pollen sac. Now, other than nourishment, what are the other things it does? It also does, students, it helps in releasing some enzymes. That enzymes will be actually used in microsporogenesis, like your enzyme like Calase. Voice is breaking? What is wrong here? Okay, nourishment mainly. For me, it is okay. For other people, just let me know. To do nourishment for the growing pollen sac. Other people, just let me know if voice is breaking or not. Just check your internet, students. Mine is working properly. Yeah. That's what I told you in the starting of the session, when I am turning to the board, because this is pulling, right? This is. Wait 1 second. I'll just untangle this. Maybe because of that. Oh God, it's so tangled, students. I don't think this tangling will happen. Yeah, it got untangled. Okay, now it's fine. A little more better. Okay, it's because when I'm moving, right? When I'm moving, it's breaking. Now, like when robot, how will I teach? How will I stand and teach like a robot? Should I call someone to fix it, or is it fine? Chalo. Okay. Now not audible. Yeah. So when I move that side, this side, this little problem will come. Otherwise, we are good to go. Okay. Okay. Fine. Students, this one helps in nourishment, also releases some enzyme. They have dense cytoplasm with more nuclei. Okay, they have dense cytoplasm with more nuclei. Yeah, so when I move, that's it. Okay, got it. Okay, so I'll try avoid talking when I'm turning. So students, this tapetum has dense cytoplasm with many nuclei. Okay, they have dense cytoplasm with many nuclei. Now the most catchiest thing about this tapetum is that they are polyploidy in nature. Okay, polyploidy in nature. They are showing polyploidy. This was the question that was asked. Okay, this was the question they asked about tapetum's ploidy level, and they asked about which is the layer where the cells will be short-lived. So two questions you got from this previous year in PYQs, that is about the middle layer and about the polyploidy. Is this clear? Okay. Yes, is this clear? Okay. Students, also this tapetum has a dense, that means DNA is more because they are forming different enzymes, right? They're forming different compounds. The DNA also, the amount of DNA also is little increased in the tapetal cells. Okay, increased in the tapetal cells. Done and dusted. So all the 4 layers we have written everything about these layers. Okay, it was an easy job to write this box. Now all you have to do is study the box. Okay, if I draw like this, yes, study the box. Okay, students, we have more to learn about tapetum. Okay, we will learn it. Yes, done. Okay, let's go to the next topic. Sporogenous tissue. No, we are going to get into directly microsporogenesis. After that, I'll tell you about sporogenous tissue. So students, microsporogenesis, before we read into this, I will explain you, then we will read this. Okay, then only we'll understand. Chalo. Now microsporogenesis, we understood that, okay, this many layers are there in anther, anther is protected, all this we understood. Now what is microsporogenesis? Okay, microsporogenesis, yes, pollen kitten, all that will come later. Pollen kitten, all that will come later. Now you understand, microsporogenesis means I have to form pollen grains, right? Now I just have some tissues with me. I just have microspore mother cell with me. Now I have to make sure that pollen grains are formed, because what is the ultimate job of the anther? Ultimate job of anther is to form pollen grains. If that only is not done properly, then what is the point? So microsporogenesis is when we convert or form with the help of microspore mother cell, when we form pollen grains. Okay, when we form pollen grains, that is what is happening. Okay, don't take screenshot, I'll send it to you people. Okay, don't take screenshot, I'll send it to you people. Is this clear? Okay, chalo. So students, sporogenous tissue word you will see a lot of places. Some places you will see that sporogenous tissues undergo meiotic division and form the microspore tetrad. That means microspore mother cell will be some places ignored. Okay, but let's write it this way. Your sporogenous tissue matures to form your microspore mother cell, or your sporogenous tissue undergoes meiotic division, form your microspore mother cell, MMC. This microspore mother cells will form your microspore tetrad, yes. Okay, microsporogenesis means what? Okay, yes or no? Okay, so students, this sporogenous tissue, each cell in the sporogenous tissue is a potential microspore mother cell. We can tell it that way, right? We can tell it that way. We can tell this sporogenous tissue is a cell compactly, homogeneously compactly packed cells. Each cell has a potential to become a microspore mother cell. This microspore mother cell will undergo meiotic division to form microspore tetrad. Fine, this is what I am saying. Okay, microspore tetrad. Fine. Now if I draw this, if I draw these structures, how will they look like? say this is your sporogenous tissue, okay? Many cells are there. Each cell has the potential to become an MMC, microspore mother cell, okay? From here to here, meiosis has happened. This microspore mother cell formed microspore tetrad. Okay, microspore tetrads.
Did you tell them Crystal and Garfield?
No, I did not. Okay, yeah, tongue twister as usual, exactly. Microspore tetrad, okay. Students, now this microspore tetrad, no, if I draw it like this, maybe you will not understand. I think we can draw it like this, but yeah, I'll show you how it will be drawn. We can draw it like this also. Okay, this microspore tetrad completely is packed. What do I mean by packed? They're covered like this, they're attached with each other. Like this. They're not allowed to separate. They're not allowed to separate. So what will we do? That is because they are covered. They are covered, or they are kept together because of one person. Okay, one person. To break this person, right? To break this person, we will get one enzyme which is released by your tapetum. Now, how many of you can answer this? Let me see how many of you can answer this. I had told this before. Okay, so students, there is one person called as callose. Okay, one person called as callose. This callose will make sure that the microspore tetrads do not go everywhere. They stick together. All the 4 tetrads will be together. But what, can I do that? No, I cannot do that, because I need each of the microspore tetrad to form a pollen grain. If they become like a group, my work will not be done, right? Then no use of microsporogenesis. So to remove this callose, we are going to take the help of our hero, that is tapetum. Tapetum is going to release callase enzyme to break this callose. Now after breaking, I'll be able to get individual microspore tetrads. So far good? Yes or no? All of you tell me in the chats. Yes, okay, all of them. Now we got free microspores, okay. Now free microspores, each of them will mature to form your pollen grain. Each of them will mature to form your pollen grain. Okay, with 2 layers in this. Yes, this is your microsporogenesis. Okay. Now let's write here. Oh, no. Relax. Done? Good to go? All of you, any doubts so far? Callase, callose, understood? Callase, callose also you understood? Yes or no? All of you, quickly tell me. Yes, ma'am, understood. Okay, done. Now students, you might be thinking, oh ma'am, this callose, no, is such a problem. Because of this callose only all these microspores kept together. But understand students, this callose is also actually a help. They provide an isolation barrier for this microspore tetrads. Okay, they provide an isolation barriers, like how we take care about a fetus, that means your baby, right? How we take care of a baby, how our fetus is protected inside the womb. Same way, this callose enzyme makes sure that there is an isolation barrier between the microspore tetrads and the other sister tetrads and other things. Okay, fine. So here you can see Sporogenous tissue was there. It became, after meiotic division, MMC happened. MMC gave rise to microspore tetrads. This microspore tetrads were attached together. Okay, they were binding together with the help of, that binding was because of callose. We used the enzyme called callase enzyme to remove the binding. After that, we got free microspores. Okay, we got free microspores. Each microspore will end at the end of the day form your pollen grain. Pollen grain. Okay. Now callose, what did I tell? Callose is going to do isolation. It's going to provide that isolation barrier. Done. Are we good to go? Fine, done. I'm not writing meiosis 1, meiosis 2. Okay, but if you want to write, you can definitely write it. Okay, you can definitely write it. But otherwise, everywhere I'm writing meiosis. Okay, now we will go back to our NCERT and we'll see if we understood or not. Look here, students. In a transverse section, a typical microsporangium appears near the circular in outline. Okay, that is this butterfly shape, right? They are circular in outline. It is generally surrounded by 4 wall layers. What are the 4 wall layers? Elephant eats my tomato. That is EEMT. The epidermis, endothelium, middle layer and the tapetum. The outer 3 walls perform the function of protection and help in dehiscence. Any line I have missed from NCERT in my notes? Tell me yes or no. Yes or no? No. Rasna, first your microsporangia. Then it will later mature to pollen sacs. Inside the pollen sacs, your pollen grains will be there, right? This sporogenous tissues, this each cell will be potent to, or potential, they'll have the potential to form microspore mother cell. Microspore mother cell. So, pollen, don't get confused with pollen sac and don't get confused with pollen grain. Okay? Pollen sac is a bag, like where you keep the rice, like rice sac. Inside that, rice is there. Formation of rice is different, sac is different. Clear? Don't get it confused. Fine? Okay. Now the innermost wall layer is the tapetum. It nourishes the developing pollen grains. Cells of the tapetum possesses dense cytoplasm and generally have more than one nucleus. Okay, they have more than one nucleus. Can you think of how tapetal cells could become binucleate? Now, you keep thinking, okay, when the anther is young, a group of compactly arranged, see, very important, a group of compactly arranged homogeneous cells called as sporogenous tissues occupies the center of the each microsporangium. Okay, center of each microsporangium. Microsporogenesis. Now, let's see what's microsporogenesis. As the anther develops, the cells of the sporogenous tissue undergo meiotic division to form microspore tetrads. So, are you understanding now, Rasna? Understanding what we are forming? Okay, from the microsporangium that was there, in between there was sporogenous tissue. That tissues had the potent to form microspore tetrads. This microspore tetrads ultimately forms the pollen grain. So, later in the pollen sac, you will be able to see the pollen grains. Okay, chalo. What would be the ploidy of the cells of the tetrad? Again, questions over questions. Now, look here. As each cells of the sporogenous tissue is capable of giving rise to a microspore tetrad, each one is a potential pollen or a microspore mother cell. Understood? Each of them, we can say, see, directly from sporogenous tissue to microspore tetrad, you can say, or you can say sporogenous tissue, MMC, microspore tetrad, pollen grains. Okay? They will form, each of them is a potential pollen or a microspore mother cell. The process of formation of microspores from a pollen mother cell through meiosis is called as microsporogenesis. Students, don't get confused with PMC and MMC. Both are the same only. Okay, through meiosis is called as microsporogenesis. Clear? Okay, microsporogenesis. The microspores, as they are formed, are arranged in a cluster of 4 cells called the microspore tetrad. As the anthers mature and dehydrate, the microspores dissociate from each other and develop into pollen grain. So, here they are not mentioning about your callase enzyme, but questions have come previously about callase and callose. Okay, that's why we are mentioning it. Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with the dehiscence of anther. So, we have to wait for anther to get dehiscent to release this pollen grains, right? If the anther dehiscence does not happen, then the pollen grains will not be released. Pollen grains will not be released. So, here dehiscence should happen, right? Dehiscence should happen. Dehiscence of what? Dehiscence of anther should happen. Clear? Clear? Microsporogenesis, let's quickly revise. So there are some types of tissues or cells called as sporogenous tissues which are accumulated in the microsporangium. Okay, microsporangium, you will heard, no? Microsporangium, between the microsporangium. In that, cells have the potential to become a pollen mother cell or your microspore mother cell or form microspore tetrad. Each of them will undergo meiotic division, form a microspore tetrad. After this microspore tetrad is formed, they will be attached together. So we will use the calase enzyme to remove this and make them free microspores. These free microspores will end up forming your pollen grains. This pollen grains will be scattered on your stigma for fertilization to happen. That's all, students. That's all. Okay? Okay? I don't like callose, it seems. Ayyo. Students, drink water. Don't forget to drink water. This weather will tell you not to drink, but you should drink. Yes or no? Chalo, let's do pollen grain now. Now we have to do pollen grain, okay? Microsporogenesis is clear. Now we have to do pollen grain. Chalo, let's do pollen grain. See, pollen grain. Pollen grains can be of different size, shape. You know, some looks like your COVID, right? This looks like your coronavirus. So they can be of different size and shape. Now, what makes them so adamant, what makes them so special is the presence of the outer layer and the inner layer. So students, if say this is your pollen grain, okay, a big pollen grain I'm drawing, and they have an outer layer and an inner layer. Okay. Okay, this is how your pollen grain looks like. Got it? This is a more beautiful pollen grain, but this much beautiful might be there, I don't know. Yes. Now let's start labeling this. Understand, pollen grains have two layers. Okay, one outermost layer, one inner layer. Okay, one outermost layer and inner layer. So if I write about pollen grain, I should know that they have two layers. Two layers. The outer layer, okay, the outer layer is called the exine. What is it called? Exine. And the inner layer is called intine. Inner layer is called intine. Now students, the outer layer is so strong, okay, so strong that still now we haven't found anything that can damage this exine. Why so? Because this exine is made up of sporopollenin. Yes? Yes? Yes or no? Okay, so this outermost layer, so pollen grains have two layers, exine and intine. Exine is the outermost layer. Exine is so strong, students, it is made up of sporopollenin, which still now we haven't found anything that can damage it, like any acid, any alkali, heat, nothing has ever damaged the sporopollenin. It is that strong. Why have they given such a strong material? It's obvious, students, because pollens get scattered to every place, right? Wind scatters them, animals scatter them, many other means like water scattering is happening. So these will go and land in any condition. It could be in a desert, it could be in a cold place. So we have to make sure that this pollen does not get damaged, because this is a male gamete. How can we let it damage? Now inside this we have this orange color which I have drawn is your intine. Okay, intine. Intine is the inner layer, okay, which is not that strong and complex like a sporopollenin, but intine is also important. Okay, yes, that's why we can store it for a very long time. Perfect. Yes, totally agree. Okay, totally agree. Okay, clear? Okay, chalo. Now intine, students, is also made up of something called as cellulose and pectin. Okay, cellulose and pectin. The intine is made up of cellulose and pectin. Understand, this pollen grain has around 25 to 30 micrometer big. That means kuti small one. Okay, but I have drawn this big. 3-cell pollen grain. Why? Yes, tell me. 25 to 30 arrow, 25 to 50. Okay, 25 to 50 micrometer. It will be micrometer. That will be the size of your pollen grain. Okay, size of your pollen grain, 20 to 50. 20 to 50 micrometer. Exine is made up of sporopollen, intine is made up of cellulose and pectin. Now, if you see here, whenever I drew exine, I left small gaps. Those gaps are called as germ pore. Okay, what are these gaps called as? Germ pore. Why do I need germ pore, students? I told you, sporopollen is such a hard material, it will not allow anything to pass through it, right? It will not get damaged at all. Then how will the pollen tube formation will happen? Only if the pollen tube formation happens, it will be able to reach all the way till the ovary. So to form that pollen tube, we have to leave these germ pores empty. Okay, we have to leave this germ pores empty. Is this clear to all of you? Yes or no? All of you tell me in the chats. Clear, easy, no? Pollen grains are very easy. Exactly. Germ pore is a place where the sporopollen will be absent so that the pollen tube can germinate. So, I am writing it here. This is the place where your pollen tube will germinate. Pollen tube will germinate. Okay, pollen tube will germinate. Done? Yes, awesome. Clear, ma'am. Okay, clear. Okay. Now, let's write few more things about this exine and intine. Students, this is, as I told you, it's a very hard, rough layer. Okay, it does not get damaged. Okay, it's not easily also. Okay, cannot destroy this layer, and wherever this layer is absent, that will form the germ. This is a better, like, we can't say it's hard, it's a normal layer. Okay, normal layer, and this layer is present inner to the exine. Okay, inner to the exine. Students, understand, this exine makes the pollen grain ornamental. Okay, actually pollen grains, if you see the structure, no, they might look like you know, like how we see your what is that, desmids, how we see your cryophytes, dinoflagellates, diatoms, that beautiful structures of pollen grains will be there. That is mainly because of this exine. Okay, because this exine exhibits different ornamental structures, different kinds of, you know, patterns. So that's the reason why pollen grains, people preserve pollen grains. Okay, because they have some ornamental use. Okay, ornamental use. Is this clear to all of you? Any doubts so far? Any doubts so far? Now, if you look here, I have divided the pollen grain into two parts. Okay, I divided one top portion, one lower portion. Yes or no? What is this top portion called as? All of you tell me in the chats. What will this top portion be called as? Yes? Anyone can tell me? Is it, okay, I will not give the clue, but yeah. Anyone just give a try. Some cell, yes, cryophytes, dinoflagellates, perfect. Those is how, that's how it looks like. Okay, it looks beautiful. Now students, this top portion which I have, you know, separated, this top portion is called as my vegetative cell. What is it called as? My vegetative cell. They have dense cytoplasm. They have dense cytoplasm. They have structure which actually provides nourishment to the generative cell. Generative cell is the one who will have the male gamete. Okay, that is the one who will go all the way to the ovary. So, vegetative cell actually provides all the nourishment that is necessary for the generative cell. Is this correct? Yes or no? The lower one here is your generative cell. Okay, generative cell. This will have the male gamete with them. Male gamete with them. Yes, exactly. Lower one is the generative cell. This vegetative cell will have more cytoplasm, more reserve food materials. Why? Because it has to provide the nourishment for the generative cell so that it can reach all the way up to your ovary and form the male gametes, right? Male gametes have to be formed. So, pollen grain is clear so far? Yes or no? Yes or no? Let's see NCERT if you missed something. Pollen grains are generally spherical, measuring about 25 to 50 micrometers in diameter. It has prominently two layer walls, the hard outer layer called as the exine, which is made up of sporopollen. So nothing is missed, okay? Which one is most resistant organic material known? Like that means from our knowledge which we have known. It can withstand high temperature, strong acids and alkali. No enzyme that can degrade sporopollen. We know even to degrade our DNA we have enzymes, but sporopollen they haven't found yet. Okay, no enzyme that degrades sporopollen is so far known. Pollen grain exine has a prominent aperture called the germ pore where the sporopollen is absent. Okay, now pollen grains are well preserved as fossil because of the presence of sporopollen. The exine exhibit a fantastic array of patterns and designs. What do you think the exine should, why should the exine be hard? I already give you answer for this. Okay, what is the function of germ pore also? I answered you. The inner wall of the pollen grain is called as intine. It is a thin and a continuous layer that is made up of cellulose and pectin. The cytoplasm of the pollen grain is made up of cellulose and pectin. The cytoplasm of the pollen grain is surrounded by a plasma membrane. When the pollen grain is mature, it contains two cells, vegetative cell and the generative cell. Vegetative cell is bigger, has abundant food reserve and a large irregularly shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle shaped with dense cytoplasm and a nucleus. In over 60% of the angiosperm, pollen grains are shed in the two cell stage. I'll tell you about this, don't take tension. Okay. What happened? Okay. Yes. So, in the remaining species, the generative cells divide mitotically to give rise to two male gametes before the pollen grains are shed. So, here what they're talking about, students, let's write it here. Okay, students, here they have told some things about vegetative cell and generative cell, which again I will be teaching you. But understand one thing, your vegetative cell has more cytoplasm and they have more food reserve because it has to provide to the generative cell. Now, one statement they told, in 60%, that means if I see all my flowering plants, 60% of the angiosperm will degrade or will dehiscent in the two cell stage. That means when it is say one vegetative cell and one male gamete. But there is three cell stage, that means where two male gametes will be formed. Two male gametes will be formed. Total it become a three cell stage. That is what they are talking about. So, let's write that statement as it is because they can ask you as an assertion reasoning statement. Okay. Here what are they saying? In 60% okay, angiosperms dehiscence in two cell stage. Okay, two cell stage. That is where they have one vegetative cell, one generative cell, two cell stage, one male gamete will be there in the generative cell. But the rest 40% okay, will dehiscence in the three cell stage. That is when this generative cell will decide to form two male gametes. Any doubts, students? Please let me know. It's a little like you have to understand this, but if you understand, it's very easy. Okay. Fine, easy. So here, understand. Here the 40% dehiscence will happen in the three cell stage. That is because the generative cell will mitotically divide and form two male gametes. Okay, two male gametes. But generally 60% will get over here. Okay, where there is one male gamete and one vegetative cell, two cell stage. But here this will happen mitotically. Mitotically when the generative cell. Here only students you will study about triple fusion, double fertilization, all this you will study here. Chalo, understood? Now I want to write about vegetative cell and generative cells. Shall I write it here? We'll fit everything into one page. Okay, we'll fit everything into one page. Here I am going to write. Fine? Okay, chalo, let's write it here. Vegetative cell will have more cytoplasm, more food reserve, yes or no, and they will be providing nourishment for the generative cell. Generative cell floats in the vegetative cell. Yes or no? Perfect. Awesome. Everyone understood? Yes. But don't forget, they are spindle shaped with cytoplasm and nuclei. Okay, they are spindle shaped with cytoplasm and nuclei. Don't forget that. No place to write it here, but you can read it from your NCRT. Okay, read it from your NCRT. See here, I've marked it in your NCRT as well. Okay, here I'll just mark it. Okay, so when you get the PDF, you will not miss it out. Okay, it is spindle shaped with dense cytoplasm and a nucleus. Okay, and they float in the cytoplasm of the vegetative cell. Yes, their irregularly shaped nucleus is there in the vegetative cell. All this don't forget. 3-cell stage, okay, 2-cell stage. 2-cell stage, 60% will dehiscent, okay. 2-cell stage, 60% will dehiscent. Now look here. Pollen grains of many species cause severe allergy. Now you understood pollen grains, you understood microsporogenesis. Now our next target is to take this pollen grains to our stigma. Right, to take it to our stigma. Yes or no? So here, pollen grains of many species cause severe allergies and bronchi affiliation. Okay, students, I am not able to see that word. Okay, afflictions in some people, often leading to chronic respiratory disorders like your asthma, bronchitis, etc. It may be mentioned that Parthenium, this one, all of you would have seen this Parthenium. Yes or no? If you chumma walk around side next to your home also, you will see this growing. Before it used to grow so much, students, now it has actually reduced, okay? Because everywhere we cut and we park our vehicle, okay? Yes, thank you. Yes, okay. So, pollen grains of many species causes severe allergies. What are the allergies that they cause? Okay, what are the allergies they cause? Like asthma, bronchitis, and this is caused because of all these wild plants like who? Your Parthenium. This you would have seen very commonly, your Parthenium. Okay, yes, Parthenium or your carrot grass that came into India as a containment with the imported wheat. So this did not come, student, this is an unwelcomed guest. Okay, we did not call it, but it just came off. Okay, it just came off. So it came with imported wheat that we bought from outside that had this Parthenium. So weeds are in such a way that you don't have to be like, oh baby, you put water on it, you take put cow dung, you grow it, nothing like that. You just bring it and put it, it will do its job. It's like, okay, this is now my place, I have become the king of this place. It will start growing everywhere. Your water hyacinth also is actually not imported and all, but it is from Bengal or some place, and it was bought here, and now it has completely, you know, eradicated the entire water bodies in Kerala. Okay, many places, but especially in Kerala, it has just spread all over. Okay, so like this, weeds are like, you don't need to welcome me, I'll only come and do the job for you. So don't forget, asthma and bronchitis is the diseases that we get from pollen allergy. Now normally, like I do have dust allergy. So dust allergy is when the moment somewhere dust is there, I start sneezing. Some people, when students, let me tell you, before all the boys and girls give flower to someone, please ask them if they have pollen allergy. Otherwise, you will go with the flower, they will immediately sneeze on your face, and then story khatam. Okay, end of a beautiful story. Okay, so please ask them if they have pollen allergy or not. This is why I am teaching you biology, so that you have, okay, this one, what, depending on the situation, you should be able to behave. Yes or no? So in this AC, like, okay, I'll kill you now. Asthma, bronchitis that came to India. Okay, fine, yeah. So understood. This is why you should know biology, not just to write NEET, to use your brains, brain cells. Yes, okay. Yes, okay, understood. Fine. Don't tell anyone that I told this. Tomorrow only go and tell, okay? Oh, Gopika ma'am told like this. Okay, next is viability. What is viability? Yeah, exactly, Rasna. The AC is working very hard. AC is like, okay, I'll work more hard than you. Something it's doing. I don't know what temperature it is moving also at this point. Chalo, let's continue. All of you are ready? Okay, it has stopped now. Students, I'll give you, okay, I'll give you one small box, okay. I don't know but where to write it. I'll write it here. Huh? I'll write it here. This is actually, many students have little confusion, no? So I made a box, so I thought I'll just give you that box because yes, finish laughing. Okay, chalo, let's start. So students, some words that you get confused, I have just made a box here in my NCRT, so I'll just give it to you. See, microsporangia. This is microsporangia. All of you remember? This is one word. The other name, okay, the other name for it is pollen sac. The other name for it is pollen sac. So I'll write it on the top. This way you will never get the confusion, because actually this chapter becomes confusing because of this, when you don't have the clarity. So other name is going to be pollen sac. Okay, one clarification is clear. Okay, yes. Okay, very good, Rani. Okay, yes. Now the next one is, see, I'll give some male gametophyte. Okay, male gametophyte. This fellow will be called as, pollen grain. Yes, now this doubt is over. Okay, doubt is over. So microsporangia, which you called there, the circles that we drew, is called, will mature to form pollen sac. Same way, male gamete, what are we calling for male gamete? Pollen grain. Okay, this just write and keep it somewhere. So this way you'll never get that doubt. Okay, you'll never have that doubt. Yes, clear, pakka? Okay. If you want to write the ploidy also, you can write. Again, ploidy, here you know that here, pollen sac, this microsporangia, they have sporogenous tissue which have the capability to form your, that is your microspore mother cell, then form the tetrad, all of that is there. Again, male gametophyte, your pollen grains will be ultimately formed. That is will be N only, okay, haploid. This will be N. This will have 2N, that whole story I have to tell now. Fine? Okay, awesome, done. Chalo. Now, let's go to viability. What is viability? That means how long this pollen grain will be active and how long they'll be useful. Right? Viability, okay? Viability means what? Thank you, wonderful trophy. Thanks for giving me the trophy. Okay, so that means how long this will be active and how long fertilization can have with this pollen grains. Yes or no? Okay, yes or no? Microspore, see here, Rasna is making me drink Rasna, okay? So much work she's asking me. Yes, madam, male gametophyte, how was it formed? Because of microspore, no? So the cycle goes like this. Your microsporangia, you have sporogenous tissue inside them that will form your MMC, right? MMC, that MMC will form microspore tetrad. That will mature. Okay, that will mature to form your pollen grain. Got it? Got it? MMC, this sporogenous tissue will be present in the microsporangium, center of the microsporangium. They'll do meiotic division, form MMC, or you can directly write microspore mother cell will form microspore tetrad. That microspore tetrad will mature to form your pollen grains. Done? Okay? For people, what are you people telling? Something and all they tell. Is this clear? Students, whole story after full Ramayana, you are telling Rama is Sita's sister. What is this? Papa, Varun from that time is telling viability. Can I go to viability? People are not letting me go to viability. They're still stuck with microsporogenesis. Are we done? Are we done? Yes or no? Please understand this and let me know, then I will proceed. Otherwise, I have to explain again. Clear. Rani and Rasna, clear. Okay, who is mother, who is father, who is kid, who is everyone, understood? Full story is over, students. Full story is over. Now I'll get into female pistol. I will start now. Okay, before that let me do viability, because that Varun is, from that time he is telling the definition for viability. Okay, so viability, here we will first read this, then we'll come to, I'll write it. Okay, so when once who is shed, the pollen grains have landed to the stigma before they lose their viability, if they have to bring about fertilization. How long do you think the pollen grains retain viability? Basically, to do fertilization, they should have the ability. How long that ability will be maintained, or how long that whole composition will be maintained? That is what we are talking about. See, the period for which the pollen grains remain viable is highly variable depending on which type of plant we are talking about. Now, rice and wheat has 30 minutes, but your Rosaceae, Leguminaceae can go up to 1 month. So that difference they are talking about. Okay, to some extent depends on prevailing temperatures, humidity. In some cereals such as rice and wheat, pollen grains lose viability within 30 minutes. Within 30 minutes of their release. That means now rice is there, the pollen grains got released on the stigma. How long they can do fertilization, or how long they are viable, is for 30 minutes. After that they cannot. Okay, now after their release, and in some members like Rosaceae, Leguminaceae, Solanaceae, they maintain viability for months. Okay, for months. Now, in these cases, in some cases, we do cryopreservation. That means in minus 196 degrees Celsius, we keep this pollen grain in liquid nitrogen. Got it? Now, we will write it. Yes or no? Students, after this, we'll take one kutti break. Okay, my throat is paining, head is also paining. I don't know why. So, I'll just eat something and I'll come. Fine? I know it's not time for giving break, but still happens, students. Okay? Yes, you people only know, you will understand. Okay, chalo, viability will write. Pollen grains. Two things you have to study, two people. Okay, first one is rice and wheat. This question only will be asked. Okay, they won't ask you definition of viability. Rice and wheat up to 30 minutes. Okay, up to 30 minutes. Got it? Students, here I'll give you a trick to remember. What is the trick? Wait, I'll write it here, then only I'll understand. Roll slow. No. Rosole. Students, rosole is the trick. To think of the trick, I needed a trick. Okay, so rosole is the trick. Okay, I'm roasting myself. Yes, students, this is the trick. Rosole. In Hindi, something meaning is there for that, right? Rosole mein kya hua, something like that. I don't know. So rosole is how you remember it, or you can remember rise, shine, and learn. Okay, but I know you people will not rise or shine or learn now. So, rosole is easier. That is viability is for a month. Okay, for a month. In who? In rosole, that is Rosaceae, Leguminaceae, and your Solanaceae. Okay, Solanaceae. Okay, Leguminaceae. Yes? Done? Easy? Okay, some members of Solanaceae, Leguminaceae and Rosaceae. Not all the members, some members. Okay? Some members. Yes or no? Awesome. Now, pollen banks are there for these people. Okay, pollen banks are there. These pollen banks are basically preservation of pollen. Okay, some rare species will be there, maybe their viability gets over in few hours. So during this case, we also preserve pollen. Nowadays it's become very common, okay, because farmers are now doing cultivation. They just go to the like the agriculture place, then they will just buy the seeds and come. Before it was not like that, but now it has become very, like, everything has become very commercial. So pollen banks are basically when we preserve, okay, preserve the pollen in liquid nitrogen at minus 196 degrees Celsius. Okay, this is the pollen bank. So in pollen bank, what happens? We preserve the pollen grains in liquid nitrogen at minus 196 degrees Celsius. Okay, done? Yes or no? Seed banks, similar to seed banks. Okay. Okay. Now students, there are some pollen products. Okay, pollen products, which I'll just tell you like this only. That is your, before we go into pistil. Okay, pollen products are simple, but your like wax. Okay, all this bee wax, all this is there. No, it has become trending. It was trending during your COVID time. Now it is little reduced. Okay, little reduced because some people are very allergic to this. Many people are allergic to pollen grains. Same way, from pollen grain, products of pollen grains also can be allergic. Okay, so bee pollen or bee wax, all these are the products. Again, these questions and all will not come. Okay, but just for your information, I'm telling you. Whatever is the question will be there. Chalo, let's start the female gametophyte. So students, female gametophyte, you know that we drew your stigma, style, ovary. Okay, stigma, style and ovary. Got it? So here, students, stigma, style and ovary, you have to understand that they form the pistil. Okay, they form the pistil. Yes. So if I label this, what is going to happen? This is going to be the ovary. Ovary, style, stigma. Okay. This together is called as your carpel. Yes. This carpel is a unit. Okay, it's a unit of gynoecium. Unit of gynoecium. Yes. Okay. Now students, look here. So carpel is there. The unit of carpel forms your gynoecium is a whorl. Inside that, who is going to be there? Your carpel is going to be there. Carpel is made up of what? Stigma, style and ovary, right? Stigma, style and ovary. Inside the ovary, the most precious thing will be there, nothing but your ovules, right? Your ovules will be there. These ovules are so precious because these are the female gametophyte. These ovules are the female gametophyte. Now the pollen grain, what it has to do is, this is your pollen grain. Right? This is your pollen grain. Pollen grains will start forming the pollen tube, and this pollen tube will come all the way from the stigma up to the ovary, and it will fuse with the ovules. Got it? It will fuse with the ovules. So that is the job of your pollen grains. Now let's try to understand what is your megasporogenesis and microsporogenesis, but before that we have some few more things to study. Now students, carpels, right? Pistil, carpel. This carpel can be monocarpellary or multicarpellary. Okay, what can it be? Monocarpellary or multicarpellary. Students, I had too many classes today, so my tongue is twisting. Okay, so here it can be monocarpellary and multicarpellary. Monocarpellary means what? A single carpel. Single carpel is there. Okay, only one carpel is there. Multicarpellary means what? Many carpels are there. Now in this many carpels, students, many carpels are there. They can be fused or they can be not fused, right? They can be free. So under multicarpellary, you have to know that there are two categories again. One is fused and one is free. Yes or no? You can decide if you want to have a friend, you can decide if you don't want to be a friend with someone. So same way, multicarpellary means many carpels are there, right? Many carpels are there. That many carpels can decide if they want to be fused or if they want to be free. See, if it is fused, two examples are given in your textbook. I'll give you an easy way to learn it. The first example you're going to remember with the help of MSP. Okay, MSP. Before I go into MSP, you have to know that I cannot use the word fused and free. I have to use the word syncarpus and free apocarpus. Apocarpus. Got it? So I will use the word for fused, I will use the word syncarpus, and for free I'll use the word apocarpus. Is this clear? Now you have to remember two examples according to your textbook. Okay, first example you're going to remember as MSP. M stands for multicarpellary, S stands for syncarpus, P stands for papaver. Your poppy, papaver, somniferum, again I'll teach you in human health and diseases. So M stands for multicarpellary. Okay, multicarpellary. I'll write only multi here, okay, full I cannot write. S stands for syncarpus and P stands for papaver. Okay, P stands for papaver. Clear? Awesome. Now, free. How to remember free, ma'am? Okay, MSP you have to remember, okay? If you can remember MS Dhoni, you have to remember MSP as well. Okay, don't be like, ma'am, MSP also possible for me to remember. Now for free, you have to remember me, ma'am. You have to remember, that is M for multicarpellary, A for apocarpus, M for Michelia. Okay, apocarpus, M for Michelia. Is this clear? Is this clear? Okay, how are you going to remember? Fused, you are going to remember MSP, that is multicarpellary, syncarpus, papaver. For free, you are going to remember ma'am. Okay, ma'am is always free and fun, no? So ma'am, okay. M stands for multicarpellary, A is for apocarpus, M for Michelia. Done and dusted? Done and dusted, yes or no? More examples are there. If you want, I can write more examples as well. Yes, all of you tell me. Is it clear? Okay, now if you forget Michelia, what will you write? Rose is there, lotus you can write. Okay, rose or lotus you can write. But in your textbook, Michelia is given. So again, follow NCERT. Don't, like, multiple examples are there, students. Rose is there, lotus is there for your, that is free. Okay, again for your, which one? Your apocarpus, I told you. Syncarpus, I did not tell you, no. Syncarpus also you have your papaya, tomato, mustard, all this is there. But again, stick to NCERT. Okay, stick to NCERT. Yes? Yes, we will do questions. We will do another session for questions. Okay, not in this. In this also I have put some questions, but we will do another session for questions, Jay. Okay, don't take tension. Enough questions, okay? Why you people taking tensions? We have so much time and we have, I'm taking everyday class also. Chalo, look here. The gynoecium represents the female reproductive part of the flower. The gynoecium may consist of a single pistil, that is monocarpellary, or may be more than one pistil, that is called as multicarpellary. When there are more than one, they may be fused together or free. If they are fused together, it is called syncarpus. If they are free, it is called as apocarpus. Each pistil has three parts: stigma, style and ovary. The stigma serves as a landing platform for the pollen grains. The style is an elongated, slender part beneath the stigma. The basal bulge part of the pistil is called ovary. Inside the ovary is your ovarian cavity or your locule. You would have studied placentation. I think you are going to do morphology. So placentation, when you study, you will study locule. In each locule, one ovary is there. Right, locule you will study. The placenta is located inside the ovarian cavity. Recall the definition and types of placentation that you have studied. Students, recall, sit and recall. Okay, fine, done. Students, these are the examples that has been given here. Look here, this is your stigma style ovary, this is your syncarpus stigma. You can see all of it is fused. Then you have your apocarpus. You can see here each of them is individually separate. Okay, each of them is individually separate. And here ovule. We'll come to ovule, don't take tension. But look at the diagram. Okay, look at the diagram. Diagram is clear? Diagram is clear? That's all. Okay, that's the starting part of it. Just stigma style ovary, apocarpus, syncarpus, examples of it. Now we are going to learn ovules. So inside the ovary, ovules are going to be there. Okay, inside the ovary. So students, very basic thing to understand ovule is the number of ovules in the ovary will be equal to the number of seeds in the fruit. Agree or disagree? Agree or disagree, students? Tell me fast. All of you agreed or only one person is agreeing? Now ovule is present inside the ovary. Okay, this is our ovary. Okay, inside this locules will be there, ovaries will be present. In any different patterns they can be. Ovule is there. Now if I draw each ovule, the ovule that we normally see is called as anatropous ovule. Anatropous ovule. What is anatropous ovule? That is your chalazal end, micropylar end and the funicle. All these three, if they are coming in one line, okay, one line, that will be called as, that is not called as anatropous. That means here, I will show you in photo. See, look here. See, orthotropous means what is happening? Your micropylar end, chalazal end and your funicle is coming in a straight line. That condition is called as orthotropous. But in anatropous, what is happening? Micropylar and the chalazal end. Okay, micropylar and the chalazal end is coming in straight line, but the funicle is moving that side. Funicle is moving that side. Okay. Yes or no? Clear, students? Did you all smash the like button? All of you? Come on, we can do this. Yes, horse shaped. Awesome. Okay. Formula, which formula? Oh yeah, this formula, no? Yes, is equal to now. Let's come. Okay, chalo, let's draw anatropous ovule, students. So understood, anatropous ovule means simply means that the micropylar end, okay, the micropylar, okay, right, students, otherwise I won't get place to draw. Micropylar end, okay, the chalazal end and the funicle are not in straight line. Okay, not in straight line. This is your anatomy. Okay, everything has maths and physics in it. Formula is there. Why, students? Why? You people tell me. Okay, why are they torturing biology teachers who left maths? Okay, now if I have to draw anatropous ovule, see, I can draw it in two ways. I will try drawing it in one way at least. Okay, I will try to draw it in one way at least. Why are you so sad, hero? Yes, hero is sad for me, students. You people are not sad, hero is sad for me. Okay, so I will try to do the anatropous ovule. I think it's gone wrong, but let's try. Okay, let's fix the issues that we make. Okay, no, not bad. Little golu molu it looks because nicely I have given food, but otherwise it's not this big. Okay, yeah, it's not this big also sometimes. I'll try to fix it as much as possible. Okay, now it looks nice. Yes or no? Okay, done? Fine? Okay? Little okay? Adjust, okay? Little okay, it should come. Okay, chalo. Let's label this part. This is your anatropous ovule, okay? Gopika ma'am's ovule, basically. So here, I'm going to draw few things. Okay, wait. To look like, chalo, done. Perfect. Okay, perfect. Now, let's draw the micropylar end and the chalazal end. Micropylar end is here. Okay, where through which the pollen tube will enter. Chalazal end will be opposite to the micropylar end. Okay, so this is going to be micropylar end. Micropylar end. That is, students, through where the pollen grain will be allowed to enter. Okay? Oh, Hindi and all is coming. This is called the chalazal end. Chalazal end. Okay, now you can see 2 layers I have drawn. Yes or no? 2 layers I have drawn. Okay, that 2 layers, thank you students for hyping me up for all the diagrams that I drew. Okay, the 2 layers are, students, nothing but integuments. Okay, integuments. They're basically for protection. Okay, integuments. Okay, there's an outer integument and inner integument. Both are going to do protection itself, but we have to give them name, outer, inner. Okay, now hilum is there. This spot is called hilum, where the funicle is attaching to the ovule is called as the hilum. You have hilum in kidney also, okay, don't forget hilum. Then inside this is the main, the heroine. Heroine is protected in multiple layers, students, but before that one more layer is there. Let's draw it with some light color. Okay, one more layer. This is the nucellus. Okay, this is the nucellus. It will be coming here. I don't like destroying my diagram. Wait. All of you wait until I draw. Okay, this is the nucellus. Okay, this is, why is the color not shown, students? This is the nucellus. It's okay, in your this one the color will be shown, okay, in your notes. Okay, nucellus. And this is our heroine, embryo sac. Inside the embryo sac, your egg is there. Okay, embryo sac, egg is there. Here filiform apparatus will be there. Later I'll show you. Now no need to crowd it. Is this clear? Let me draw all the end. Wait. Chalazal end, nucellus, embryo sac, integuments, hilum, micropylar end. Students, funicle we forgot, students. Papa, who is holding all the burden of the family? Funicle. Funicle done. Nucellus done. Embryo sac done. Integuments done, no? Everything done. Just check your NCRT. Yeah, just looking like a wow. Okay? Don't tell such things, okay? I'm a biology teacher. Don't make of zoology teacher. Students, anything I missed, tell fast. I'm looking at your chats. I can't see this far, so now I have to look here and here. Okay? What and all happens in old age. Tell fast. Clear? Yes, ma'am. Okay, for Pavitra, it's clear. Education is crying. Okay, education, don't cry. It's okay, happens. In life, more important things will be there. What should I answer, Mahendra? Chalo. Okay, got it. Done. Okay, this is your anatropous ovule. Now we have to learn the each job of these people, right? Who are these people? Why are they here? First, we'll start with funicle. Funicle is like the stalk. Right, funicle is like a stalk that holds this entire ovule in place. Funicle is just like a stalk that holds the entire ovule in place. Next, if I go to micropylar end, this micropylar end is the one that shows the pollen tube. That means it will tell the pollen tube that you can come here because filiform apparatus will be there. That filiform apparatus will guide, telling that, hey, you come this way. We tell, no, our parents will give one on our head and we'll be like, come this way. Okay. Students, your senior is here. Please, all of you stand up and respect your senior for 1 minute. All of you. He is sitting in the class and he is telling these things don't work, okay? Because he is, okay? So, please, all of you stand up and students, my tolerance level is less. So, I removed your senior from the class, okay? Yes, but please give respect. Okay, next time you see him anywhere, please give him respect. That's what I have given. Integuments both are there, outer and inner integuments both are there. Okay, chalo. Now look here, students. This micropylar end is the end through which the pollen tube will enter. This is showing the direction. Okay, this is showing the direction. Yes, and then hilum is the place where the funicle, where the ovule and the funicle attaches. Okay, this spot, can you see? Here is the attachment. You can see that the funicle and the ovule is attaching with each other. That is the hilum. Okay, that is the hilum. Then integuments, there are outer integuments and inner integuments. Both are going to do basic protection. Okay, both are going to do basic protection. Now, embryo sac is the place where your egg is there. Okay, embryo sac is the main heroine. It is kept inside. Inside that only egg is there. That egg has to fuse with your pollen grains to form the fertilization. Done? Chalo. Then nucellus. Students, nucellus are a type of cell which actually gives the food reserve. It has the food reserve to give it to the to the embryo sac. Like how tapetum was there to, you know, how tapetum was there to give it to the pollen grain. Same way you have nucellus cells which will give the food reserve for the embryo sac. Okay? Yes? Micropoil is not have. What they don't have? Filiform apparatus job is to guide the pollen grain. Like come this way, don't go that way. Come this way, don't go to the antipodal cells. Come to the synergids. So that is the job of them. Okay? Chalo. Done. Now students, what is that? Okay, fine. Then you have the chalazal end. Chalazal end is just opposite to the micropylar end. Okay? Done and dusted. This also understood. No, you only have to know orthotropous and anatropous. Again, hemianatropous, which is like this. That is your micropylar end. Chalazal end is in straight line, but the funicle is perpendicular to it. Then you students, this are all no need students. It's all unnecessary. Okay. Chalo, let's do megasporogenesis. Now, is this clear? Okay, is this clear? Okay, to guide the pollen tube. Yes, perfect. Embryo sac you understood. Now our job is to form the egg. Right? How do we form the egg? How do we do the megasporogenesis? Right? Let's do that. Are you all with me on this? Did you all smash the like button? Telling my throat, I'll become of old. Okay, now we have to form the female gametophyte. For that we are going to take the help of megasporogenesis. Going to happen, students, you have megaspore mother cell. Okay, you have megaspore mother cell, MMC itself. Okay, this MMC will undergo meiotic division and form micros, that is megaspore tetrads. Okay, it will form megaspore tetrads. All similar only. Okay, megaspore tetrads will be formed. But here everything is mega. Okay, megaspore tetrad. But one problem is there. Megaspore tetrad is formed. But this megaspore tetrad, 3 of them will degenerate. 3 of the cells will degenerate. This 3 will degenerate. Only one, this fellow, he will become functional. Not fellow students. Okay, this one person, girl, will become functional. I don't know what is the, I turn it is breaking, now it will be fine. When I face like this, it will be fine. When I turn, it will break. Okay. So students, what is going to happen? Yes, only one megastar is there. That will become functional. That will become your functional megaspore. Okay. This is going to be your functional megaspore. Other 3 of them will degenerate. Other 3 will degenerate. Got it? Now, still breaks, huh? Don't know what's the issue here. Wait, I'll call. When I face like this, it will be fine. When I turn, I don't know why this issue is coming. Wait, 1 second, 1 second. Okay. Huh. Yes. So, functional megaspore. Yes. So good. So far, so good. Okay. One became functional. Okay. Only one becomes functional. The rest will get degenerated. Is this clear? Chalo. Okay. Now students, this one megaspore functional megaspore, they will have a 2-cell stage, 4-cell stage, 8-cell stage. Finally, this 8-cell stage will arrange themselves to form the embryo sac. Okay, embryo sac. Fine now? Okay. Okay. I think it's only when I keep it inside. What and all problems in life. Okay, so this functional megaspore will have a 2-cell stage. Okay. Okay, 2-cell stage. Look at NCRT, you will see the same diagram. Then, okay, a 4-cell stage. This 4-cell stage will become an 8-cell stage. And this 8-cell stage will ultimately form an embryo sac. Will ultimately form an embryo sac. Yes or no? Agreed all of you? Agreed all of you? Okay, there will be only one functional megaspore. This again students, it will undergo around 3 times karyokinesis. What is karyokinesis? That means division of the nucleus. Okay, around 3 times karyokinesis will happen. See, this is not mentioned in your NCRT, that's why I didn't want to focus much, but there will be some students who want to learn. So, it will form 3 times karyokinesis and it will go from 2-cell stage to 4-cell stage, 4 to 8, and finally form the embryo sac. This entire embryo sac will be 7-celled 8 nucleate stage. 7-celled 8 nucleate stage. Got it? 7-celled 8 nucleate stage embryo sac will be formed. So far good. So far good. Okay, megasporogenesis clear. Okay, 7-celled 8 nucleate. This cell will be considered as 1. The central cell or the polar nuclei will be considered as 1. Okay, fine. All of you understood? Fine. Now let's see what NCRT is telling. The megasporangium, let us familiarize ourselves with the structure of a typical angiosperm ovule. Okay. Now, the ovule is a small structure attached to the placenta. Locule is there. Okay, this is the locule. This portion is the placenta. Through the placenta, the ovule will be attached. Wow, nothing can be seen. My pen also is not seen. Something colorful I have to take, students. Wait. Oh. Okay. So, this is the locule. Okay. This portion, this cap is the placenta. With the cap, the ovule will be attached. Okay, ovule will be attached. That's how you will study your parietal placentation, marginal placentation and all that. Okay, chalo. Now, the ovule is a small structure attached to the placenta by the means of a stalk called as funicle. I told you, this is a basic stalk which holds the entire ovule in place. The body of the ovule fuses with the funicle in a region called hilum. Exact thing I told you. Okay, now, thus the hilum represents the junction between the ovule and the funicle. So, we can tell funicle is here, ovule is attaching like this. This junction is called as the hilum. Okay. Now, each ovule has 2 protective envelopes called the integuments. There is an outer integument and an inner integument. Okay. Integuments encircle the nucellus except at the tip where there is a small opening called as the micropyle. So, wherever there is an opening, understand that pollen tube has to go through that opening. That is why that opening is there. Otherwise, why will that opening be there? Opposite to the micropylar end is the chalazal end, which represents the basal part of the ovule. Enclosed within the integuments is a mass of cell called nucellus. Cells of the nucellus have abundant food reserve. Okay, located in the nucellus is the embryo sac or the female gametophyte, and ovule generally has a single embryo sac formed megaspore. So, understand, embryo sac is the female gametophyte, not the female gamete. Okay, female gametophyte. Got it? Yes? Clear? Now, let's see what is megasporangium. Now, students, here megasporogenesis is the process of formation of megaspores from the megaspore mother cell is called as megasporogenesis. There what was is the process of formation of microspore from the sporogenous tissue or from the microspore mother cell is called as your microsporogenesis. Got it? Ovules generally differentiate a single megaspore mother cell in the micropylar region. Now look here, you will understand better. Now students, look here. This is your nucellus, which is actually giving the food reserve. In middle, you have the megaspore mother cell. This megaspore mother cell will undergo meiotic division. Okay, it will undergo meiotic division and become like this. Okay, it will become like this. That means it will start forming a megaspore dyad. First it will form dyad, then tetrad. Okay, then first dyad, then tetrad. Okay, all this time micropylar end, you can see this is nucellus. Okay, continuously providing the nourishment. From the megaspore tetrad dyad, it became megaspore tetrad. But no use. 3 of them will degenerate. 3 of them will degenerate and only one will become functional. Okay, only one will become functional. Now look here, that one which got functional will have 2 cells, 4 cells, 8 cell stage. That is why we tell 3 times karyokinesis is happening. That means 3 times the nucleus is dividing. Okay, nucleus division is happening. In the 8 celled stage, they will start arranging themselves. They will start 3 cells will start moving towards the pole. Okay, 3 cells here, 8 cell is there. Okay, 3 cells will start moving towards the pole. This 3 cells will be antipodal cells. This 3 cells will be 2 synergids and 1 egg apparatus. Again, I will teach you that. Now, this is an embryo sac. So, let's draw an embryo sac. Where is the slide? Okay, let's draw an embryo sac. Then we will be able to understand. Okay, so this is the embryo sac. So before this, there is one stage, that is, can I duplicate this? Okay, in this stage, what is happening? This is the 8th cell stage. Okay, this is the 8th cell stage. Now we are going to draw the embryo sac. Okay. Got it? Students, when I draw, it will look more beautiful because I am drawing like flowers petal, but it's not this nice also. Okay. Chalo. Now we have one more person to draw, who is the guiding light of life. Okay, done. Embryo sac done. Okay, yes. Let's label it. Students, the cells which are present on the chalazal end, okay, the cells which are present on the chalazal end, there are around 3 cells. These 3 cells are called as antipodal cells. What are these called as? Antipodal cells. Got it? Antipodal cells. 3 of them will be there. Okay, all are like sisters, 3 sisters, trio. Okay, in the middle, what you see is called as the central cell. You can tell polar nuclei. This is considered as one entity. That is why it is called 7-celled 8-nucleated state. Definitely they have an individual nucleus, but they will be considered as one cell. They will be considered as one central cell. Got it? Yes. Very good. Now in between I have drawn one green color, big one. This becomes the egg. This is the egg. This becomes the female gamete. This whole embryo sac becomes the female gametophyte. If you do not understand this difference, you will never understand this chapter. Understand who is the gametophyte. Gametophyte means the one that is bearing the gamete. Okay? And the gamete means which will actually interfere in the fertilization and form the zygote. Okay? Now students, 2 pink color structures I have drawn. That will be the synergids. Synergids. This together will be called as the egg apparatus. Egg apparatus. That means 2 synergids plus 1 egg will be called as the egg apparatus. Done? Clear? Embryo sac is done. Any doubts? Done. Okay. Now here I drew some worm-like structures, no? That is your filiform apparatus. Filiform apparatus. Students, this filiform apparatus is the one who guides the pollen tube to enter. Because how will the pollen tube come? They'll come like this and they'll enter to the egg apparatus, right? So this filiform apparatus will guide, okay, will guide the pollen tube. Guide the pollen tube entry. Done? Done? Yes? Yes. This is the micropylar end. Okay, I mean this is the chalazal end, the top one. Here. On the chalazal end only your antipodals will be there. Because here there is no opening for the tube, that is your pollen tube to enter. Here only there is the opening. Okay, done? This is your embryo sac. Fine? Yes, perfect. Guides the pollen grain. All of you? Yeah, it's filiform. Okay, filiform apparatus. Filiform apparatus, okay, not fully formed, filiform. Okay, got it? So students, when this formation is happening, no, here, when you see these formations, here, see, these formations will happen mitotically. From 2-cell stage to this one, all of you know, no, mitotically it will happen. Mitotically it will happen again from here to here, here to here. Here meiotically it was happening, okay, here, because here division have to be formed. Here it will happen meiosis 1, meiosis 2 will happen. Here mitotic divisions will happen, okay, mitotic divisions will happen. So today we are going to learn pollination. All of you would have heard in your smaller classes pollination. That is very simple, students. So this is your stigma, right? This is your stigma. So stigma is there, anther is there. Okay, in this anther some pollens will be there. Yes or no? Pollens will be there. The anther will dehiscent and this pollen will fall on the stigma. So pollination means what? Transfer of pollen grains from anther to the stigma. It can be between same plants, it can be between different plants. All of you are with me on this? Okay. So what is this? Pollination meaning that transfer of, okay, transfer, can you see here? Transfer of pollen from where to where, students? From anther to stigma. Anther to stigma, right? Stigma is the landing, like how your airport is there, no, where the flight will land. Same way stigma is the landing where the pollen grains will come and land. Okay, so that is why this structure is given, this kind of a structure is given. Now the pollen grains will be here, right? The pollen grains will be here, will come and settle here, and then later pollen tube formation, other things will happen. So the first ever step is they have to meet, right? Who has to meet? The male gamete has to meet the female gamete. Do you people understand? Right? Yes. So transfer of pollen grains from anther to the stigma of a same plant or of a different plant, okay? Of a same plant or different plant. Okay. Now actually students, I can divide this pollination into different types, right? As I told you, they can be same plants, they can be different plants. On the same plants, they can be on two different flowers. So it's like however they want, they can do it. So I'm going to divide pollination into three types, okay? According to your NCERT, I'm going to divide it into three types, and I'm also going to tell you the common names of it. It's very easy. So, if I divide pollination, okay, if I divide pollination, students, any extra point I'm telling, you have to make a note in your NCERT, okay? Later, don't struggle telling you where is ma'am's notes, which is the class, because I'll take 2-3 revisions of this chapter on the channel. So you will be confused, where did ma'am tell? Okay, so look here, pollination I'm dividing into three types. First one is called as autogamy. First one is called as what? Autogamy. Second one is called geitonogamy. Geitonogamy. Third one is called xenogamy. Ma'am, what is this gammy and all you're telling, right? Why can't it be simple, right? Why can't it, these names be like Rani, Hasini, but it's not, okay? It is going to be more than that. That is autogamy, geitonogamy and xenogamy. Auto means what, students? The moment someone stands and we'll be like, okay, I'm putting the phone in auto mode. That means automatically the brightness is controlled, contrast is controlled, lighting is controlled, frame is set, focus is set. The phone does everything by itself. So auto means self, okay? Auto means what? Self. Is this clear? Yes, very good. Auto means self. So that means this is self-pollination. This is self-pollination. But all of you think, if self-pollination has to happen, that means, don't you think the anther and the stigma should be in the same flower? That means this flower has to be bisexual. All of you tell me, if the flower has to be bisexual, anther and stigma should be in the same plant or on the different plant? Self, everything is self. They're like, hey, I don't want to be dependent on you. Who are you? So they're completely independent, okay? Independent, full, I don't know, students, empowerment is happening. All of you tell me the answer for this. Does the flower has to be bisexual? Yes, the flower has to be bisexual, because the pollen, that means the anther and the stigma, has to be in the same flower for self-pollination to happen. So the condition is that the flower has to be bisexual. Bisexual. They are independent. They are independent. Okay, full financially well settled family there, okay? Yes, well settled family there. Exactly, like your hibiscus or your China rose, they tell, no? So self-pollination, bisexual, and they're independent. This is the main condition of your autogamy. But there is one biggest problem in autogamy. What is that, students? Now imagine you're taking everyday photos in your auto mode. Their phone is only doing, okay, phone is only adjusting, but sometimes you want photos in such a way, like, you know, you should look very amazing, right? You should impress everyone on your Instagram, Facebook, that kind of a photo you want to take. That means you will have to do some kind of editing, you will have to set up something. Same way in this auto mode, every time, because you're taking in auto mode, there is no variation, right? There is no variation. So even in the self-pollination, the main problem is that there is no variation. There is no variation. Students, variation is not there. All of you know, like your theory, right? Strongest. The strongest will survive. So for that also variation should be there, because the nature is going to select. Yes or no? Yes, awesome. So no variation is the biggest problem of autogamy. Now when you come to geitonogamy, what is this geitonogamy? Then you have xenogamy. So let's take geitonogamy. Students, geitonogamy is very simple. This is a flower. Okay, autogamy has some more types. I'll teach you that. Geitonogamy, imagine this is a plant. Okay, this is one flower. So no color only. Wait. Yeah, this is a flower. Okay, this is one more flower. And let's say here one more flower is there. Okay, these are the 3 flowers in this plant. Okay, now let's label these flowers also. So imagine this is the male flower, female flower, and this is also a female flower. Now what has happened? This is one plant. Agreed, ma'am, it's on one plant, multiple flowers are there. So from here the anther has to go. That means the pollen has gone and fell on this female flower. That means on the same plant, on different flowers, the pollination has happened. So geitonogamy is when transfer of pollen grains from the anther to the stigma of different flowers on the same plant. Are you all getting this point? Are you all getting this point? Yes? Awesome. Okay. Yeah, biggest problem. I think many of you haven't marked your attendance. Please mark your attendance, students. I'm waiting for all of you to mark your attendance. Don't be such lazy people. Mark your attendance, all of you. Smash the like button, all of you. Do one basic task I give you every day. Okay, so gatenogamy is that transfer is happening, but not on the same flower, on the different flowers, but on the same plant. Okay, same plant. Clear? Chalo. Now xenogamy. Students, xenogamy, if I tell you in simple words, it means cross pollination. Cross pollination. Okay, cross pollination. Here what is happening? Here variation will be there. Okay, variation will be there. That is the biggest thing. Okay, variation will be maximum. Now what is happening here? It's very simple. This is one stigma. Okay, this is one stigma. This is an anther. Okay, of a different plant. On this anther, there is going to be pollens. That pollen will come and fall on this, right? But this is a plant by itself, this is a plant by itself. Okay, got it? So they are on different plants. They are on different plants. This is the flower. Okay, flower, students. Do you see the art and the artist? Okay, see, ma'am know to draw botany diagrams also well. Yes or no? Tell me in the chats. Okay, this is one flower, that is one anther, that is another on another flower. Yeah, done. Okay, done. Okay, done. Exactly. Like your papaya. Okay, so that means they are on different plants and they are going to do cross pollination. Because they are on different plants, variation is going to be maximum in xenogamy. So xenogamy is simply called as cross pollination, which you have studied in your smaller classes. Got it, all of you? Are we all good to go? Now, in autogamy, students, there are some more types. In autogamy, there are some more types. Okay, so we are going to learn these types. Oh no, I was not supposed to do this. Wait, students, let me check. Okay, 1 second. Ma'am did something very beautiful. Okay, 1 second, students, let's go here. Okay, autogamy has some more types. All of you tell me in your NCERT it is given. NCERT it is given. Under autogamy, they have given you cleistogamy and chasmogamous. Yes or no? Cleistogamous, right? Cleistogamous and chasmogamous. Now, who are these two people, students? Why did they give it under autogamy? That means they have something to do with self-pollination. Yes or no? Yes or no? They have to do something with self-pollination. All of you are agreeing with me or not? Okay, yes. Yes. Okay, so cleistogamous flower means they are always closed, students. They don't want to open. They don't want to open. They don't want to expose. They want to be the introvert that they are. They don't want to open. So these flowers are closed. Okay, they are closed flowers. Now, in closed flowers, the problem is that you cannot expect in closed flowers cross-pollination to happen. Yes or no? How can you expect something to be completely closed to do cross-pollination or like to do that crisscross and have variation? Not possible. So, this cleistogamous flower has only one option, that is to do autogamy or to do self-pollination. Okay, to do autogamy or self-pollination. All of you are with me on this, yes or no? Yes, all of you? Awesome. Yes, awesome. Only autogamy in cleistogamy. Yes, 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 yes. That looks like a rhyming word, no? Autogamy in cleistogamy. Okay. Yes. Now, students, here if I say chasmogamous, they are open sometime, closed sometime. They are both kind of a flower, but we can tell them most of the time they are open flowers with exposed anther and stigma. Okay, they are open flowers with exposed anther and stigma. You can see your NCRT. This is the exact word of NCRT. Exposed, anther, and stigma. But these flowers, no, sometimes they are closed also. So open flower or closed flower, you can see some places. Now, how do I remember the example of this flower? Cove. How do I remember the example? Cove. Tell me the full form, all of you. Camelina oxalis and voila. Yes, cove, it's cove. Okay, so they are open flowers, but sometimes they are closed flowers. So when they are closed, autogamy is the only option. Okay, they have exposed anther and stigma. Now example is cove, Camelina oxalis and voila. Students, in Camelina also, if I draw the plant, no, of Camelina, say this is the plant. Okay, they have leaves everywhere. Now if I draw the flower bud, okay, if I draw the flower bud, say this is your chasmogamous flowers, which are open sometimes, but in the same plant, no, there can be clistogamous flowers. They'll be completely closed. So this Camelina can be example of both, okay, chasmogamous and clistogamous. So these flowers which are completely closed will be to the lower end, and these flowers are your clistogamous flower. Okay, clistogamous flower. And these one on the same plant will be the open one, that will be chasmogamous. Chasmogamous. Yes or no? Understood? All of you? Clear on this? Okay? Okay, awesome. Clear? Perfectly clear? Perfectly clear? Yes. Perfect, ma'am. Let's move on in life. Yes? All of you smash the like button quickly. I'm waiting for you all to smash the like button. If no, then I don't know what to even do. Okay, I cannot even scold you people at this point. You're all grown ups. How much will I scold you? Okay, so done. So students, this is all about your pollination. Yes or no? So we understood that pollination means transfer of pollen grains from the anther to the stigma of the same plant or on the different plant. Then we have autogamy, where no variation happens, and this is self-pollination, happens in bisexual flowers, and it is completely independent, okay, because it's happening in the same plant. Then if I come to geitonogamy, here what is happening? Transfer of pollen grains is happening from on the same plant but on the different flowers. Okay, if you want me to write, I'm writing it here small. Okay, this is happening on different flowers. Okay, now I'll ask you all a question. In geitonogamy, how much do you think high variation will be there? Okay, high variation will be there in geitonogamy. All of you try to tell me. Thank you, wonderful trophy. Geitonogamy, do you think high variation will be there? How exactly? I told you, see, in some families, you know, they marry Mama Punna, we tell, right? Mama Punna, Mama Payyan, they tell. That is, dad's sister's son, you get married, something like that. Okay, now imagine instead of dad's sister, you got married to someone else in the family itself, like dad's brother's younger son, kuch bhi, something you get married like that. That means you're still in the family. You're not going out of the family. Just because you're not marrying that person, that Mama Paya or Mama Punna, that does not mean that, you know, big variations will happen. Still, the diseases that is running in the family will continue. Okay, so no much variation will happen. Please do not write. They might ask you a question in assertion, reasoning form, statement based question asking geitonogamy shows much variation because it's happening between two flowers. Your answer should be not at all. Okay, variation will be very, very slight, sometimes negligible also. Okay, I want all of you to answer. Only I can see few of them answering. Okay, so no much variation. No much variation. No much variation. Got it? Chalo. Xenogamy, maximum variation will be there because it's cross pollination. It's happening between two different people. Yeah, two different people. Chalo. Okay, pollination understood. Now I told you, see, these people cannot walk, move around. That's not possible by them. So they need some agents to do this, right? They need some agents to do this. So the agents of pollination means the people who will actually help them in pollination. So we will divide this agents, okay? Agents of pollination into two types, okay? Mainly two categories. Two categories. I've kept your NCRT in front of me, so don't take tension. I'm looking at NCRT. I'm seeing if I've missed anything. Nothing has been missed. I'm looking at it and then doing it, okay? So don't be like, ma'am, missed this. Don't take tension. NCRT is right in front of me and I'm looking at NCRT and I'm seeing if there is anything that is missed, okay? Because sometimes when we teach a lot, no, we have chances that, okay, we forgot some small, very important thing. So that's why we all teachers keep NCRT in front of us. Okay, biotic. So students, I'm dividing the agents of pollination into abiotic and biotic. Abiotic means what? The non-living part. Okay, biotic means what? Something that is living. Simple as that. Now, under abiotic, I want to divide them into two categories, two main categories, 1 and 2. First one is wind, second one is water. First one is wind, second one is water. What is that?
Hot water.
Oh. Students, Baswa sir is here. Please come, sir. Just tell them hi. Yeah, come.
How's it going, ma'am?
It's going good.
Oh my god, it looks like a Rin, no? Rin the ad again. I'm not even visible there.
Student, sir is glowing with the t-shirt because of the lights,
Exactly.
right? Hello, tell hi.
Hi. I told hi, right?
No, I'm telling them to tell you hi.
Reverse.
Reverse.
Yeah, reverse.
Yes, in all NCRT, in all lectures.
NCRT lecture is covered. Nice. In all NCRT lecture is covered.
You know, recently I typed them, like the smash button. They're like,
I feel the same. So many times it happens, right?
they're like, ma'am, we cannot see the smash button on YouTube. Awesome.
Yeah.
Okay. Yes.
You continue, ma'am.
Yes, sure. Thank you, sir.
Feels so nice.
Okay, so students, abiotic we are divided into, that is two categories will be wind and water. Okay, wind and water. Now in biotic, only one you have to study. Who is that? Insects. You have to study only one person, that is insects. Okay, but the task is not to remember wind and water. You have to know the other name for it as well. Okay, other name for it as well. Done? All of you? You lazy people. Yes, 1 second, students. Yesterday was captain's birthday. People didn't wish. Very bad. Yesterday was captain's birthday. Chalo. Students, now wind, water, insect we have, okay? Now you have to know how these people are different. Understand? Now, example, I'm asking you a question. Wind has to, when the wind is blowing, it will take some pollen grains with it and deposit on some stigma. Now, does wind actually care if the flower is showy, if the flower is colorful, if they have nectar? Nothing. Wind that does not matter. At the same time, water. In the water, you know that the amount of pollen grains that is going all the way, there are chances that the pollen grains get lost. The chances that the pollen grains does not reach the stigma. So they will need an exposed stigma or a feathery stigma so that the pollen grains get attracted. At the same time, even for the wind, okay? But if you see insects generally, insects get attracted to colors. Insects get attracted to flowers which are showy. So even if the flower is small, they will try, the nature will try to make it an inflorescence. That means like a bouquet of flowers, right? Where even the smaller flowers will arrange themselves in such a way that they will look like a bouquet so that the insects get attracted. And insects also care about the nectar. If this flower has nectar, then the honeybees, those people are going to get attracted to this. So that is kind of like a reward that this insect gets for doing pollination. Is this clear to all of you? Yes, it can be nectarless, odorless when it comes to wind and water. All of you are with me on this, I hope? Yes, ma'am. Okay. Now, students, as I told you, in names that you have to remember is, this is anemophily. Okay, anemophily. When it is done by wind, it is called as anemophily. When it is done by water, it is called as hydrophily. I'll write water here. Okay. Yes, it is called as hydrophily. Done? When it is done by insects, all of you tell me. I taught you last class also. Entomophily. Entomophily. We tell entomology, no? Entomophily. Yes, all of you are with me on this? Very good, Madhumita. Yes, very good. Awesome. Yes. So here we fall under insects. Now, not only insects, see, we have some garden lizard. Okay, garden lizard also helps in pollination. We have rodents, we have bats, we have birds. For birds, it's separate name. For bats, it's separate name. But according to NCRT, only insects you have to study. That is entomophily. Done? Awesome. All of you are answering? Perfect. Chalo. Let's move on. All of you? Okay, now I have put some NCERT snips as well, so I will show you with the NCERT snip, but before that I'll teach you just one little extra information, but it is required, okay, because it is mentioned in NCERT but name is not mentioned, so you cannot skip it, right? They will be like, okay, the teacher was supposed to teach you, so just make a note in your NCERT itself. Very good, ma'am. Entomophily, ma'am, entomophily. Yes, Jay has answered. Yes, Rani has answered. Okay, okay, very good. Very good. Okay. Students, now hydrophily I want to divide into two, okay. One is called as epihydrophily, okay, epihydrophily, and hypohydrophily, hypohydrophily. Now, ma'am, what is this epihydrophily and hypohydrophily? Just by the name itself, you will be able to understand, yes or no? Okay. Now, students, epihydrophily means it occurs over the surface of the water. Okay, imagine there is plants. Okay, like let's say Vallisneria is there. Okay, this Vallisneria, the flowers are on the top. So, if the pollination is happening above the surface, it will be called as epihydrophily. If it is happening below the surface, like your seagrass, okay, Zostera, it is happening below the surface. That will be called as hypohydrophily. Okay, this is mentioned in NCERT, but the name as such is not taken up multiple times, so that's why I am going to make it right. Okay, chalo. So epihydrophily means occurs, okay, occurs above the water surface, above the water surface, okay. Hypohydrophily means below the water surface. That means below the water surface. Okay, yes, all of you? Yes, very good. So hypohydrophily example is, example here, first this, here example you have your Vallisneria, okay, water hyacinth, multiple things are there. I am writing only one example, okay, and Zostera or your seagrass. Done, students? Done? All of you? Is this clear? Any doubts on this? Okay. Any doubts? Any doubts? Ma'am's color skills. Okay, this is your 3 things that you have to remember very importantly. Now I have put NCERT. Okay, now I have put NCERT, so we'll study from NCERT. Okay, yes, below the water surface. Very good. Okay, chalo, let's learn NCERT. So pollination by wind. Students, in pollination by wind, water and insect, you have to study what is the type that they're showing. Like, do they have feathery stigma? Pollens are like this. That is what you have to focus, not about just the name. Okay, name is not important. You have to know the, you have to focus on the how these people are. Okay, pollination by wind is more common amongst abiotic pollination. That means if I categorize all the abiotic factors, in all the abiotic factors, wind is the most common. Question can come, so keep it in your mind. Okay, wind pollination also requires that the pollen grains are light, right? They have to be lightweight. Imagine I have to carry this and go. It has to be lightweight. If it's heavy, it's not going to be carried. It's going to be stuck on the plant itself. So first ever character is that pollen grain are light and non-sticky. They do not have to stick. They shouldn't be sticky, right? So that they can be transported in the wind currents. Okay. Now, they often possesses well-exposed stamens. Can anyone tell me why is this so? It is understood that if they don't have a well-exposed stamen, then there are chances that the pollen will not be scattered or the wind currents will not be able to take the pollen if the anther is completely downwards and it's not well exposed. Okay, chalo. Okay, yes, so that the pollens are easily dispersed into the wind currents and large, often feathery stigmas. Okay, large, often you can see feathery stigmas to easily trap the airborne pollen grains. So stigma also has to be feathery. Just by pollen grain, oh, pollen grain is coming in the wind. Stigma is not even seen. Then that also won't happen. No, stigma also has to be feathery so that it can hold the pollen grain. So both of them have to be active people. Okay. Yes. Wind pollinated flowers often have single ovule. Okay. They have single ovule. Wind pollinated flowers often have single ovule in each ovary and numerous flowers packed in an inflorescence. Okay, packed in an inflorescence, because main thing is that they are not showy plants. Okay, the wind does not care if the flower is showy or not. The just main goal is that they should have an exposed anther, and the stigma on which this is going to fall should be feathery so that they both can attract each other. But imagine sometimes flowers can be small. So what they will do? They'll keep an inflorescence, so maximum pollen gets scattered. So the probability of pollination to happen will be maximum. Okay, chalo, they will be packed in inflorescence. A familiar example is your corn cob. The tassels you see are nothing but the stigma and the style which wave in the wind to trap the pollen grains. Wind pollination is quite common in grasses as well. Wind pollination is quite common in monocots as well. Okay. Then anything else? Anything else? Wonderful trophy? All nonsense. Yes, students? Okay. So, corn cob, the tassel you see are nothing but stigma and style which wave in the wind to trap pollen grains. Wind pollination is quite common in grasses. Okay. In monocots, this is very, very common. Okay. Chalo. So, students, what are the characteristics of your wind pollinated? Very simple. Let's revise once again. The pollen grains. First ever thing is that the pollen grains have to be light and non-sticky. Okay, light and non-sticky. Now, how should the stigma be? The stigma should be feathery so that it can catch the pollen grains. Okay. At the same time, the flowers usually will be in an inflorescence. Then, did I miss some other point? Did I miss some other point? Tell me quickly. Light, non-sticky, feathery stigma. Inflorescence, flowers should be in an inflorescence. They have to be exposed, the anthers have to be exposed, right? That's it. Example, monocots, most of the monocots do that, and other corn cob, right? Corn cob, and wind is very most common, abiotic kind of pollination, right? Light and non-sticky, done. Okay, chalo, now water pollinated. Some examples of water pollinated are Vallisneria and Hydrilla. Okay, Hydrilla, which grown in freshwater and several marine seagrasses such as Zostera. Now, not all aquatic plants use water for pollination. In a majority of aquatic plants such as water hyacinth and water lily, the flowers emerge above the level of water and are pollinated by insects or wind, as most in common plants. So, students understood? Most of them, what they do, they come above the surface, which is called as epihydrophily, where they come above the surface and they wait for insects or the wind to pollinate them, because the chances of water is quite rare. This water hydrophily is quite rare, and only 30 genera of plants are doing this kind of water hydrophily. Because the main problem is, students, the pollen grains have to be transferred through the water. It has to reach the stigma. Compatibility is there. So it's a process where the probability of pollination to happen is very less. So plants need to do pollination, right? That's the whole process. So they will come above the water, which is called as epihydrophily. Okay, do not forget, the flowers emerge above the surface of the water and are pollinated by insects or wind in the most of the land plants. In Vallisneria, the female flowers reach the surface of water by a long stalk, and the male flower or the pollen grains are released on the surface of water. So the water currents are going to take this pollen grains to the Vallisneria. At the same time, this Vallisneria the flower is going to come up with the help of a long stalk, waiting for the pollen grains to be carried by the water currents. Chalo. They are carried passively by the water currents, okay? By the water currents. Some of them eventually reach the female flowers and the stigma. In other group of water pollinated plants such as seagrasses, so this is going to be hypohydrophily, okay? Female flowers remain submerged in the water. Very important, students. Lot of statement based question has come from this Vallisneria and Zostera or your seagrass. So keep in your mind, okay? Keep in your mind. Chalo. Okay, in water, and the pollen grains are released inside the water. So this whole process is happening submerged. But at the same time, in Vallisneria, what is happening? Everything is happening above, but the pollen grains are coming through the water current. That's why they are falling under hydrophily. Now, pollen grains are released inside the water. Pollen grains in many such species are long, ribbon-like, and they are carried passively inside the water currents. So they have to be long and ribbon-like so they do not get lost. Okay, if they're small like one small one like one sand piece, they will get lost definitely. So some of them reach the stigma and achieve pollination. In most of the water pollinator species, pollen grains are protected by a wetting, that is by mucilaginous covering. Obviously, because the pollen grains shouldn't get soaked, right? So they have a mucilaginous covering so that the pollen grains does not get completely wet, or they sink down. All this to avoid many things. Students, underwater, what and all things happen. And students, around 30 genera, okay, around 30 genera only does your hydrophily, very less. And all of you should also know about your pteridophytes, bryophytes, pteridophytes and your gymnosperm. And this is the main reason why bryophyte is called the amphibian of the plant kingdom. Why is it called amphibian? Because it cannot complete its life cycle without water. Same in amphibians, they need both land and water. So they cannot complete the whole cycle without water. So that's why we call bryophytes as the amphibian of the plant kingdom. Okay. So this bryophyte, pteridophyte, gymnosperm also, some of them do this hydrophily, or at least in their one stage they will need this water for pollination. Yes. I hope that was clear. I have told that from the NCERT. Okay. Yes. Majority, now we'll go to insect pollination. Majority of the insect pollinated flowers are large, colorful, fragrant and rich in nectar. These are the floral rewards that they're getting, okay? The rewards, I'll write, okay? The rewards that these plants get for doing pollination is the nectar, okay? The nectar. This is one big thing, big reason why the insects come. Right, your honeybees also, they wait for the nectar. So, fragrant and rich in nectar. When the flowers are small, a number of flowers are clustered into an inflorescence. So, students, this is very normal thing, okay? That means whenever the flower is small, they do some kind of modification, like the bract will be modified. Some kind of modification will be done so that the insects get attracted. This the plant itself will do, okay? Now, to make them conspicuous. Now, animals are attracted to flowers by color or by their fragrance. So, this nectar, same way, fragrance also attract these insects very, very, okay, fragrance. Now, it need not be always good fragrance also. Now, if you see your Rafflesia, Rafflesia has a bad odor. Okay, it has like a dead something dead. Okay, dead and decayed smell is there, but that will attract the flies, because flies always, if you have seen there is a dump somewhere, the flies will be there, right? That's why we tell, do not allow flies to enter your home, because it will immediately cause your entamoeba, histolytica, all of you remember this, amoebiasis and all that. So that's because these people get attracted to dead and decayed smells. So that's the main reason, okay? So Rafflesia flower has a foul smell, but still flies get attracted to them. So majority of them want good fragrance and nectar, but then there are some of them who would want the foul smell as well, okay? Now, animals are attracted to flowers by their color and fragrance. The flowers pollinated by flies, beetles secrete foul odors, okay, to attract these animals. So, understood? It's the butterflies, the bees, all of these people get attracted to the nice smell and nectar, but then there are also flies and beetles who get attracted to foul smell. Example, you can remember Rafflesia. They will not ask you example because it's not there in NCERT. To sustain animal visit, the flowers have to provide rewards to the animal. Nectar and pollen grains are usually the floral rewards, okay? Nectar and pollen grains. These are the rewards that is given to your people, telling that students, now in your home if a guest come, first time you gave them nicely one chai, you were like, okay, I'll get you people some tea, I'll get you some like bhaji, bhaji, bonda, then I don't know, samosa in some sites. So they'll be like, okay, next time also let's go there. Next time also let's go there, right? Because they treated us so well. But the next time they come, you're like, oh, these people came again. You will not give them properly. The third time they will not come. If they still come, then you have to question their self-respect, but otherwise they will not come, okay? They will generally not come. By your second visit, if they see your face changing, not giving them properly, sugar is not there, tea powder is not there, milk is not there, you tell all that, then they'll understand, okay, first time they were very sweet, this is their actual color, okay? So even the insect will understand. First time the flower has given them nectar, pollen grains, they'll be like, okay, next time also I'll visit. Next time something they do, some jugaad, some gadbad they do, then they'll be like, hey, you go. There are 100 other flowers, I'll go to them. Okay, so Flowers also, see, ego is there, students. Everything on earth has some ego level, okay? Egoistic people are. See, flower also, insect also, okay? Insect also. Yes, so these are the usual flower rewards. Okay, done. Any doubts so far? Please ask me. Any doubts? Any doubts? Any doubts? No doubts? All perfect? Did we miss something? Yes, we have to do mutualism, but otherwise, tell me, just take NCERT and tell me. In my opinion, nothing is there, okay? Now we only have your mutualism. Mutualism. Okay, clear? Okay. Sorry students, I have got a very good cold. Okay. Yes. Chalo, let's do mutualism. Since mutualism, you have only 2 examples, but there are more examples actually. Generally, if you see, there are more examples, but you have only 2 examples. First one is your Amorphophallus. Okay, and then you have your Yucca. Okay, now what is this is, these plants have their ovary. In the ovary, they will allow the moth to lay egg. Now what is happening? Moth is getting the space to protect their egg, and also this flower is getting a free pollinating agent as well. So this is called as mutualism. So this way what is happening students, because the main problem that insect faces, if you see butterflies also, the main problem that they face is if they lay the eggs, the chances of the eggs being, you know, undisturbed without being caught inside the predator's eyes is very difficult task. But if these plants are providing the ovary for them to lay the eggs and protect them, then they'll be very happy. So this mutualism happens, that is, I am helping you, you help me, we both will be benefited out of it. All of you got this? So that is why they have written this, okay? And this one is a 6 feet tall plant. Students, very much taller than us, okay? 6 feet tall plant. So the chances of this plant getting affected also is very less. Other plants will get, you know, ego complex. No, my God, such a tall plant. Yes? Yes, places, yes, it provides safe place for insect to lay the eggs, okay, as the floral reward. So it will give that safe space safe space to lay eggs for the insects, okay, insect as a floral reward. Then I'll put it in arrow. Students, you will get all these notes on Vedantu NEET English Telegram as well as on Biology by Gopika. So do not forget to join the Telegram, okay? If you want all these notes, there is no other place where you will find the notes. The link will be put in the chat later, but not now. Immediately, if you want these notes, immediately after the class, I'll share the notes on the Telegram. So download the notes and keep it, so you will have a folder. Okay, later when you're doing revision, these notes will be the most helpful notes, but I would still recommend you to write, okay, write at least 1 sheet where all the important words are there. See, now these 2 words, there are chances that you forget, because you have studied so much theory, there are chances that you forget this. So these kind of things, write and keep it. Okay, write and keep it. Yes, very good. Okay, so floral rewards will be given. Now Yucca and your Yucca moth, that is your Prunuba. Okay, such a nice name, no students? Prunuba plus Prunuba is the moth name, okay? Prunuba. This is the only Indian thing that I've seen in the entire sexual reproduction in flowering plants. Okay, so these people also are the same thing. Okay, one is providing the ovary, safe space for laying the eggs. Safe space for laying egg. The other person is doing what? Okay, acting as a pollinator. Students, for them also, like matrimony, tension is there. Ki will I get chosen? what if the insect does not come, what if pollination does not happen, then my survival, my existence in this world will be questioned. So they also go through depression. So what they will do, they'll be like, hey, you come and lay egg, you do free pollination for me. So pollinator also they will get free. Okay, so do you understand? Everywhere problems and standards are set. Okay, yes, done students? Yes. So these are obligate mutualisms. Okay, now this is actually a very simple thing. Ovary is there, they will lay the eggs, protection is done, at the same time they get a free pollinator as well. Done? All of you? Energy is there? Can we move to the next topic? Yeah, put some fire emoji in the chat. What, you all are sleeping? I am sick. Why are you people sick? Come on, all of you. Okay, fine, little energy is there. Okay, let's go to the next topic, students. Next topic is the most important topics that we are going to do with. That means, like, what do I mean is, the questions from these portions are more. Okay, questions from these portions are more. Now, okay, you are a very self-content person. That means the plant is like, okay, I'm not independent on anyone. I have the stigma, I have the anther, pollination will happen, self-pollination will happen. I do not want dependency on anyone. Okay. Now the nature is like, no, I will not let you to be in this much peace also, that you're so self-content that you feel everything is proper in your life. I will not let you do that. So the nature itself decides that when an plant, okay, when they continued self-pollination, continued self-pollination, the nature will be like, you're going to have depression, bro. Okay, so he'll tell, nature will be like, no, I won't let you be like this. You should also face the problem. So inbreeding depression is going to happen. Okay, inbreeding depression. That means when the plant undergoes continuous self-pollination, the plant will be like, okay, I am in depression. So inbreeding depression will happen. So this is a kind of, this is the game, because if inbreeding depression did not exist, then even variation will not exist. Yes or no? The self-pollinated plants will be very happy. They are completely independent. They will not even require anyone's help. So automatically no variation also will happen. Yes or no? Right? No variation will happen because they are very self-content. Okay, they are very self-content. So nature is like, why should only plants have fun? No, why should only, cross-pollinated plants have fun? You should also have some fun. So you will get inbreeding depression. Now, to solve this inbreeding depression, outbreeding devices has come, students. So this whole plant is like our story itself, very relatable, plants and we are. Okay, so even when we sit alone for a long time, we'll be like, okay, there is some problem with me. I need to talk to people, right? I'm going into depression. So same way, plants also will go into depression. So let's see what is this outbreeding devices, okay? Okay, now outbreeding devices means to avoid inbreeding depression, or to, basically to block this self-pollination from happening, we have this outbreeding devices. Are you all following, or is it only Jay who is following my class? Only Jay seems to react. Where are the other students? Come on, students, you have to be active. It's like I'm talking to the camera and you people are least bothered. Okay, you have to talk to me. Who else will I talk to? I'll talk to the walls, huh? Students, inbreeding depression will happen. Students sitting inside the studio and talking alone. Have some karuna. Yes, okay, done. Ready? I'll do outbreeding devices. If not, I will not do. Okay, first solution. First problem is that this self-pollinated flowers have bisexual flowers. So nature told, let's do one thing. Let's make it unisexual flowers. Okay, let's make unisexual flowers. Let's make unisexual flowers. Like example, your papaya. Okay, they told first solution for avoiding self-pollination is let's make unisexual flowers. Okay, let's make unisexual flowers. Is this clear to all of you? Okay, many examples are there, students. You can write papaya, you can write date palm, examples. Okay, so unisexual flowers. Second solution that they gave was, let's do one thing, let's do something called as changing the position of anther and stigma. So look here, all of you, this is your stigma. Okay, this is your stigma. Okay, this is the anther. Okay, this is the anther. Yes, this is the anther. Now, it is very understood, right? Very common, okay, that the pollen grains from this anther will definitely land on the stigma. Where else will it go? Where else will it go? Because the stigma is down, anther is above. So, obviously, this pollen grains will fall on the stigma. So, now they told, okay, let's do one thing, let's change the position of this whole setup only. We'll change the whole setup. The second setup they made was simple. Why should boys have all the fun? So the girl became tall. Since whatever I'm telling is for joke, okay, so that you remember for a longer time. They made this setup. Okay, they made this setup. What is the setup that they made? Simple thing. They lowered the anther's height. Okay, they lowered the filament which was a long stalk, right? They lowered the size of that and they increased the size of stigma. Now you tell me, how will pollen grains land? They cannot land, right? They cannot land. So this is what? Okay, so this is what they did. Basically, they changed the position of the anther and stigma's placement. Are you all with me on this? Yes, all of you, different positions of anther and stigma. I'm going to write. Okay. Yes, lowering anther's height. Exactly. Okay, so this happened, that is heterostyly. Okay, heterostyly. This is where different position of anther and stigma. Okay, changing the position of, I'm writing it down, students, changing the position of anther and stigma. Okay, anther and stigma. Okay, heterostyly. Then heterostyly, it's called heterostyly. Okay, yes. Next one, let's do one thing. Let's make it asynchronized. Now they both are in very much synchronization. That means pollen falls on stigma, stigma gets compatible, then pollen tube formation happens, germination happens. Let's do one thing. Let's make them asynchronized. Okay, that means they will not synchronize with each other. So we will make it okay, let's make it asynchronized. Okay, all this is done to avoid self-pollination. Okay, yes, Karthik, totally agree to you. Okay, yes or no? Okay, so we will not make them synchronized. Now, what do I mean by synchronized, ma'am? That is, here we call it dichotomy, but you don't have to take tension of that. That is, imagine pollen and stigma, right? Pollen also matures at the same time, forms the pollen grain. Stigma also matures. The ovule is ready. Okay. Now the egg is ready inside the embryo sac. We will do one thing. We will make it the maturation or the rate at which they both are maturing. Let's make it asynchronized. This way, even if the pollen matures first, the stigma is still not matured. Egg is not formed. So how will they get self-pollinated? So we will do asynchronization. Is this clear? Is this clear to all of you? Please make a note of all this. Very, very important. Okay, very, very, very important. Dichogamy. This condition is called dichogamy. Even if you don't remember this, it's okay. It's okay. Okay. Yes. You have protandry and all, but then again stigma released first. Yes, they are not matured at the same time. Okay, they are not matured at the same time. Okay, not matured at the same time. Yes, done. Protogyny and all you don't need to know now, but if you know it, good and well. But then questions will not come because it's not there in NCERT. No, name is not there. Name is not there. Okay. Yes, not matured at the same time. Yes, correct. Perfect. Ansh, Arya, perfect. Protogyny, you don't have to know now, right now. Okay. Protogyny, that is basically if pollen is maturing first, is different name. If anther is, stigma is maturing first, is different name. Okay. Chalo. Next, the last solution. What do we do, students? What is the last solution that we do? Okay, we did unisexual flowers, we did heterostyle, now we did dichogamy. Now the last thing we have to do, something else also we have to do. Who can tell me the answer for that? Yes, we'll make them do this self-incompatibility. That means from the same flower, if the pollen is falling, the stigma will tell, okay, you're not compatible to me. So we will do self-incompatibility. Okay, we will avoid self-pollination. Okay. Self-incompatibility. Got it? Yes, sure. Students, climate is changing, can't do anything. It's very common. Yes, self-incompatibility. Okay, I'll draw a diagram and show you that. But where do I draw the diagram is a question. Okay, students, I'm taking it here, all the way here. Okay, here like this. Now imagine this is the stigma. Okay, this is the stigma. This is the stigma. Imagine pollens has come and fallen here. Okay, pollen have fallen here, here. Okay, 2 pollens have fallen. Now the self-incompatibility, I'll make sure that the pollen tube germination will not happen. Okay, pollen tube, the same pollens will not germinate. So here what is going to happen? I will make this pollen, only maybe this will germinate. Okay, this will germinate and form the pollen tube. So this way what will happen? Self-incompatibility will be maintained. Yes, done. So the main 4 outbreeding devices according to your textbook, there are around 6 of them, but you don't have to know. Out of the NCERT one, only 4 you have to know. First one is formation of unisexual flowers, because only when bisexual flowers are formed, self-pollination chances are more. So we will form unisexual flowers, example papaya, date palm, etc. Second one is heterostyle, where the anther and stigma position will be changed. Before, anther will be above so that the pollen grains will directly land on the stigma. Now what did we do? We raised the size of the stigma. The style got increased. This way the anthers have come below. Now the pollen grains chances to reach there is very, very less. Okay. So that one. Third one is asynchronizing. That means both of their maturation time will be changed. Okay, pollens will mature first, stigma will mature later, or vice versa. Depending on that, they have their particular names as well. Okay, that is dichogamy. Now not mature at the same time. Next is self-compatibility. Okay, self-incompatibility. Did I write compatibility? Okay, self-incompatibility. That means the same pollen, the self pollens will make sure that the germination of the pollen tube does not happen. This way what will happen? Again, self-pollination will be hindered, and the one that came from outside, right, cross-pollination, that pollen will be allowed to form the pollen tube. So this way we will make sure that the pollen tube germination does not happen with the same pollen. I hope this is clear. 4 points, please keep it in mind. Outbreeding devices, lot of questions have come. Done, students? Is this clear? Yes, can I proceed? Yes, ma'am, you can proceed. Yes, okay. Students, we still haven't reached the 100 likes. So sad, yeah. So sad. I think you people don't want classes anymore. Yeah, you people don't want classes, huh? Yes? Okay, chalo. Let's do one very, very interesting topic, pollen pistil interaction, double fertilization and triple fusion. Okay. Now students, as I told you, now wind is bringing the pollens, water is bringing the pollens, insect is bringing the pollens. But see, understand, this insect, wind, water, they are not Amazon delivery people who will exactly know the location, understand that who is going to be compatible with each other and drop it there. They are just randomly bringing a random set of pollens. Now it is stigma's job to decide who is right for me, right? Even when we do a marriage also, the girl will decide, the boy will decide if this person is right for me or not, because I have to live this entire life with this person. So like that, even the plants get to choose that. That is called as compatibility. That is pollen pistil interaction. Pollen and the pistil will interact with each other and they'll get to know if they are compatible with each other or not. Okay, fine. So let's say this is your stigma. Okay, this is your stigma. I'm drawing a big stigma. Okay, this is your stigma. Okay, done. Until I draw this, all of you take your NCRT and check if did we cover everything, anything I missed. Okay, so in stigma done. Now on the stigma, different types of pollens will come and land. Okay, let's draw different colors of pollen. Now inside this, I'm also going to draw your embryo sac. Okay, a big embryo sac. This is your embryo sac. Okay, in this antipodal cells will be there. Okay, here eggs will be there. Okay, and the most important thing, your filiform apparatus. Okay, okay, done. Done, students. Embryo sac is there, stigma is there. Now let's draw different pollens on this. Okay, so the wind bought one pollen. Now as I told you, they are not going to look at the address and deliver. One more fellow bought one pollen. Okay, okay, one more pollen was bought. This color choosing only is one task. Okay, one more pollen, done. Okay, now 3 pollens have landed on the stigma. Now as I told you, it is stigma's job to select who has to go inside or who has to form the pollen tube and undergo germination. This is called as pollen pistil interaction. First step. Now imagine let's consider one is doctor, one is businessman, one is a bank manager. Okay, now first thing that what happens during a marriage, first they will talk. They'll talk and see if this person actually matches my vibe. Right, nowadays you people use the word a lot, vibe. Ma'am, he doesn't match my vibe, ma'am. Okay, this teacher does not match my vibe, ma'am, so I do not watch their classes. So that vibe check will happen. So it's not called as vibe check. Here the first thing is they will have a conversation with each other. Bank manager will tell, I will give you loan, how much ever loan you want, I'll give you. Businessman will be like, I will take you to that place, this place. Doctor will be like, okay, since I don't have time much, I'll take you to hospital and show you the hospital. So different things will be there. So that's why students, doctors are always, you know, marry doctors itself, because they will be able to understand the work pressure. Now you also will become a doctor. You will also look for a doctor itself, because you both will be able to understand the problems that you go through in the hospital. Now if you go and tell, a doctor goes and tells a businesswoman or a businessman that this is the problem that I'm facing, how will they understand, right? So first thing that they will ask, family, everyone will talk, and the first thing that is going to happen is called as chemical dialogues. That means they will both talk to each other. Okay, we call it chemical interactions. Okay, chemical interactions, or you can also say chemical dialogues. Okay, chemical interactions or chemical dialogues. Okay, so the boys will be trying to convince, who is the boy, pollen grains will be trying to convince stigma, telling that, you know, actually I am this, I am that, I am Ambani's son and all that they will tell. Okay, but the stigma will still decide. So chemical interaction happened, chemical dialogues have happened, they spoke to each other, and the stigma felt that, okay, let me do one thing, I'm ready to marry this businessman. Okay, so businessman is going to go to the next stage of, that is, okay, now the next stage will be what, students? This pollen, let's say this also got into, okay, this also got into, okay, bank manager to out, okay, now doctor also is in pending, but yeah, let's keep the doctor there, let's not, okay, scratch the doctor out. So students, now the doctor, not the doctor, the businessman will be allowed to germinate. So germination of the pollen is going to be the next step. Okay, germination of the pollen is going to be the next step. I don't think anyone will teach you in the story form. Okay, germination of the pollen. Okay, after the germination of the pollen, now what is going to happen? The pollen will start automatically forming its pollen tube, right? So let's, pollen tube formation will start. Okay, pollen tube formation will start. Can you all see that? Yes. Okay, yes, hydrating myself. Exactly, pollen germination will happen. Now pollen tube is going to emerge bigger, right? pollen tube is going to emerge bigger because it has to meet the egg. Students, girl is here, we have to meet the girl. So here, pollen tube formation. That will be the third step. I hope all of you are understanding. That means after they spoke with each other, she agreed that okay, I will choose the businessman over the doctor and the bank manager, and the next step will be where their parents and everyone will start meeting each other, no? So here what is going to happen, students? Here pollen tube formation will happen. Okay, pollen tube formation will happen like this. Okay, it will happen like this, it will happen like this, it will happen like this, it will happen like this. Okay, he is going to meet the girl. Oh, oh. Okay. Yeah, pollen tube formation is going to happen. Is this clear, all of you? So far good? Okay. Now, once they reach there, now what is going to happen? Everyone will be like, oh yeah, my daughter is going to marry a businessman. You come inside, see the house, this is that, this is this. So what is going to happen? Filiform apparatus will start calling the pollen tube, because filiform apparatus' job was to guide the pollen tube. Yes or no? So filiform apparatus will be like, come inside, see our house. This is where I used to stay. This is where Didi used to study. So now she'll give introduction to what was happening in the house and basically embarrass the kid. Okay, embarrass that girl who's going to get married. So this filiform apparatus will guide. Okay, will guide pollen tube entry. Pollen tube entry. Yes or no? Now they are going to meet. Okay, now they are going to meet, and this pollen tube will start moving inside. This pink color which I have drawn is your filiform apparatus. Okay, yes, exactly. Guide the pollen tube. Got it? Okay. But one problem happens, students. The problem is that in the starting, it was in a 2-cell stage. Okay, here it was in a 2-cell stage. 2-cell stage means what? That, okay, one vegetative cell is there, one generative cell is there. All of you remember pollen grain, no? So here, at this position, they were in a 2-cell stage. Okay, 2-celled stage. That is, one vegetative cell, one generative cell. But as it moved down, as the pollen tube formation happened, somewhere here, it underwent a mitosis. I'll use another color. Okay, it underwent a mitosis. Mitotic division. Okay, division happened. During this mitotic division, it formed a 3-cell stage. It formed a 3-celled stage. In this 3-cell stage, one vegetative cell and 2 generative cells were formed. 2 generative cells were formed. Here what happened? The businessman also came. The doctor is like, okay, now I am not going to give up. Now even I want to get married. So the doctor also will come inside. Doctor also will come inside. One will get married with the egg. Not married, student. One will fuse with the egg. Okay, another one will fuse with the polar nuclei. So here what is going to happen, as the mitotic division happens, when they reach the 3-cell stage, this vegetative cell will completely degenerate. Why? Continuously giving nutrition to this 2 generative cell. Okay, 2 generative cell. Yes or no? Okay, students, because here, see, from 2-cell stage, it is forming 3-cell stage. Okay, so here it is going to go through a mitotic division. Okay, so I told you this when I taught you embryo sac also. From 2-cell stage, it became 4-cell stage, 4 to 8. That time also it was going through a mitotic division. In the starting, when the MMC was going through forming megaspore, it was going through a meiosis division. Yes or no? So this you would have studied it in botany, mitosis and meiosis, right? So that is the reason here. Now look here. One vegetative cell, 2 generative cells. So vegetative cell main job was to give all the reserve food materials for the generative cell. So by here giving, this fellow got over. Okay, completed. Now 2 generative cells is there. So as I told you, one will go, the green one, right? The businessman will go and fuse with the egg. Okay, fuse with the egg. I'm not drawing this pollen tube because it will look too crowded. So I'm going to write it here. So here, this is the most important part, students. Okay, that's gone. Okay, so look here, the most important part is the businessman is gone and fused with the egg. So the businessman is going to be one generative cell fused with the egg, okay, to form what? What is the main job? What is the whole task? Voice, obviously. Okay, Sudarshan. Yes, students, the main job was what? Egg to fuse with the generative cell and form the zygote. Okay, zygote. This is the businessman and the girl. Now the doctor also came in, no, after the mitotic division. So here one generative cell will fuse with the polar nuclei. Polar nuclei. Got it? Polar nuclei is here, no? All of you remember 7-celled, 8 embryo stage. So polar nuclei it will fuse, or polar nuclei to form endosperm. To form endosperm. This endosperm is going to provide the nutrition for the growing zygote or the embryo. Is this clear to all of you? Yes or no? Yes or no? So students, this one GC plus polar nuclei, 2-cell stage, no, this is 2 here, 7-celled, 8 nuclei. So 2 nuclei are here. This is called as triple fusion. Okay, when this doctor marries the sister, no, this will be called as triple fusion. Triple fusion, okay, that is one GC plus polar nuclei forming an endosperm is called as a triple fusion. And when these two things happen in a 3-cell stage, this condition is called as double fertilization. This stage is called as double fertilization. I hope it was clear. Pollen pistil interaction, most of the students complained telling ma'am it's so difficult to understand pollen pistil interaction. Did you all understand? Students, even if you are watching it recorded, let me know in the chats, comments, if it was clear. Okay, double fertilization. Yes, because 2 fertilization happened. Now the plan was for only 1 marriage, but 2 marriages happened, so it will be double fertilization. And definitely this fellow went and fused with 2 nuclei, right? So it's going to be triple fusion, right? 3N stage, okay, 3N. So endosperm is going to be 3N. Done and dusted. All of you tell me in the chats if it was clear. Any doubts, please ask me now. Crystal clear. Okay, all of you? All of you? Perfect. Okay, done. Okay, fine, fine. So students, this is your pollen pistil interaction. Let's go through it once again. So as the pollen land on the stigma, the wind and the insect or anyone, they are not going to deliver it exactly right, because they don't know who is compatible. The stigma has to decide, right? So as soon as the pollen lands on the stigma will have a chemical dialogues with the pollen. Okay, a chemical dialogues with the pollen, and after the chemical dialogues or the chemical interaction, the stigma will decide which pollen to select. Okay, after selection of it, it will allow germination of the pollen, otherwise it will not allow germination of the pollen. Okay, the moment germination of pollen start, the small pollen tube formation will happen, and that pollen tube will be going through all the way through your micropylar end to your egg, to meet the egg. Okay, so the filiform apparatus will guide the pollen tube entry. After the guiding of the pollen tube, so in the starting it's going to be a 2-cell stage where there is 1 generative cell, 1 vegetative cell. But as it comes down, it will undergo mitosis and it will become a 3-cell stage. But also understand 60% of the anther dehiscence will happen in a 2-cell stage. This is one of the very, very important lines. All of you remember, I hope. Last class also I had told you around 60%, okay, angiosperm anthers dehiscent, angiosperm anther dehiscent in 2-celled stage. Okay, The rest 40% will undergo a 3-cell stage. Okay. Now as it comes, it will mitotically divide, form a 3-cell stage where we have 1 generative cell, 2 vegetative cell. Since this generative cell will, vegetative cell will continuously give the food reserve, the vegetative cell will be completely degenerated by the time it reaches here. After that, this 2-cell stage, no students, one of the generative cell will fuse with the egg, which is the actual, like the whole plan was that, right? So one generative cell will fuse with the egg to form the zygote, and the second generative cell will go and fuse with the polar nuclei, or in the central cell, right? Polar nuclei, and form the endosperm. This endosperm will provide the nourishment for the growing embryo. Okay? Yes, is this clear? Okay, fine. Okay, this is your pollen pistil interaction, double fertilization, and your triple fusion. All the 3 concepts I have explained you in this one slide. Okay, one slide. Now, artificial hybridization. Now students, imagine we are not able to get the desired quality of plant, we are not able to get the desired characteristic. Now what we have to do? We have to do it artificially. Yes or no? So we will do artificial hybridization. That means I artificially make sure that whatever I want to get out of this plant, I will do it. So artificial hybridization means something that is done artificially. Your entire interaction, your formation of a plant, formation of whichever types you want, right, varieties you want, will be done artificially. In this you have mainly 2 things to remember. First one is called as emasculation. Okay, emasculation. Second one is called as bagging. Second one is called as bagging. Now it's very simple. Now all of us know that the agents are going to bring the pollen and dump it on the stigma. To avoid the dumping on the stigma, I am going to bag my stigma. Okay, I will cover the stigma with a bag. With a bag, students, you can also use a polythene covers. Okay. This will avoid what, students? This is going to avoid unwanted pollen pistil interaction. Okay, or you can say unwanted pollen from landing on the stigma and continuing with the germination. So unwanted pollen pistil interaction, I am writing it here. Unwanted pollen pistil interaction. Clear? First step done. I protected my stigma. Now next one is emasculation. Now I don't want this anthers also to scatter the pollens on some random stigma. So I will removal of the anther. So I'll do removal of the anther. In the stage here we are going to do removal of the anther. Are you all clear on this? Yes or no? Yes, removal of the anther is called as emasculation. Okay, removal of the anther. And what am I basically trying to do? I'm going to collect the pollen grains so that this pollen also does not get scattered unnecessary. So I am going to just do this. This is my anther. Okay, in this anther there is a lot of pollen grains. Okay, lot of pollen grains. I can collect this pollen grain and I will add it, or I will allow the pollination to happen depending on which variety I want. So collecting the pollen. Okay, so this way, this is called as your artificial hybridization. Okay, emasculation, after removal of the anther and your stigma will be bagged. After that also we will bag it and keep it so that no unwanted interactions happen. Is this clear? That's all. Emasculation and bagging, very simple, just two lines you have to know this. Nothing so difficult about it, and this is mainly helpful for the farmers. Okay, now students, we learned pre-fertilization, we learned fertilization. Now we are going to learn post-fertilization event. Okay, all of you are with me on this? Yes or no? Okay, yes or no? Students, can I take a 2 minutes break? Just 2 minutes break. I'll just get some hot water. Okay, 2 minutes break. Yes, don't go anywhere. Stay back here only. Just go through your NCRT by then. Okay. I'll be here in 2 minutes. Don't go anywhere. Ma'am is coming. Okay. Yes, students. Ready? Okay. So students, now we are going to do post fertilization event. Okay, what and all is going to happen during the post fertilization events. So let's draw a line. Okay, it looks fine, right? All of you are here? Okay, so in post fertilization events, first one is endosperm development. Okay, endosperm development. Second one is your maturation of ovary. Maturation of ovary to form fruit. Yes, all people's here. Okay, then embryo development. Embryo development. Next is maturation of the ovule to form the seed. Maturation of ovule to form seed. So these are the post fertilization events. Very simple. We form the endosperm, right? One GC, that is one generative cell, when it combined with the central polar nuclei, it formed an endosperm. Then we saw embryo development, right? Then we are going to see maturation of how ovule is going to form the seed and maturation of how ovary is going to form the fruit. So the main job for the plant to reproduce is that they are going to provide us with the fruit. Right, they're going to provide us with the fruit. So that is the whole process, right? It's not just about pollination. They are going to ultimately give us some food, right? The fruits that we consume is a kind of food. It's not only for us, even for the other animals as well, birds as well, squirrels. Many things are dependent on plants for food. Ready, students? We will start with endosperm. Okay. Now, all of us know. So students, here we have a diagram which I wanted to show you. Here all of you can see the zygote. Okay, again globular embryo, heart-shaped embryo, plumule, mature, cotyledon. This I'll show you, but just see the shapes. Okay, this is your globular embryo. That is here. You can see the globular embryo. Here you can see the heart-shaped. Okay, here you can see heart-shaped. Here globular. Okay, and then your actual radicle, all that will come. So now here. Okay, let's make. Since I have no idea how to duplicate. Okay, let's say if I do this, okay, this is gonna come. Okay, chala, no problem. Okay, no problem. We will just do it anyways. Okay, let's learn about endosperm. Students, endosperm, please understand this double fertilization is actually helpful for the embryo, because endosperm is a set of cells that actually provides the food materials, that provides the nutrition for the growing embryo. Okay, so endosperm is not something useless. They are the ones who are going to provide all the nutritional, okay, all the nutritional material for the growing embryo. For the growing embryo. Clear? For the growing embryo. Now students, if you see generally your embryo in angiosperms, in your gymnosperm, it's going to be a little different. Okay, so let's write in gymnosperm, okay, in angiosperm. I'm telling the ploidy. The ploidy is going to be a little different. Okay, in gymnosperm it's going to be haploid. Okay, in gymnosperm it's going to be haploid, and in your angiosperm it's going to be 3-end. Okay, triploid. Yes, fine. Now this is again, this is just an information. Again, sir will teach you again this in morphology of flowering plants, but as of now you just have to understand this. Even the plant kingdom you would have understood, right? Anyway, now haplontic and diplontic, that life cycle is removed, but before you would have studied this. Now students, if I divide endosperm, I can actually divide it into nuclear and cellular endosperm. Okay, endosperm, if I see the types, right, I can divide it into nuclear nuclear and cellular. Okay, nuclear and cellular. Now this words you're going to study in when I teach you about your coconut, okay? But otherwise you don't have to take tension, but understand in cellular it will be followed by cytokinesis, okay? So if I draw the division, it will be like this, okay? 2 divisions, again multiple divisions will happen. So in cellular it will be followed by a cytokinesis. That is actually the difference. I think you will study this. No, you will not study it again. But yeah, if you're taking some masters, you will understand. It will be followed by splitting of the cytoplasm, okay? So cytokinesis, okay, followed by a cytokinesis. Now nuclear and cellular. Ready students? So far good? Are we good to go? Okay, yeah. So this nuclear endosperm is a very common type, free nuclear division, okay, like your pen, okay, that is your primary endosperm nucleus. So this one is very common one, pen. Okay, I'm writing only pen here. That is your primary endosperm nuclear forming the free nucleus. Okay, free nuclei. So this is a free nuclear region. Now let's divide the endosperm according to your NCERT. Exact NCERT words we are going to write. So if I divide my endosperm, depending on if endosperm is there or not, I can divide it into 2 types. Okay, albuminous and exalbuminous. Exalbuminous. Can anyone tell me what is the difference? I told you depending on if endosperm is going to be there or not, right? Presence and absence, okay? Presence and absence, you will understand it's albuminous or exalbuminous, yes? Yes or no? Yes, Tattva is enough for NEET. Yes, definitely. If you have bought Tattva, more than enough. Okay, it has everything. It also has extra information you can filter out. Like if you don't want to learn, no problem. Okay, so in albuminous, what is going to happen, students? The endosperm will be retaining. Okay, retain some amount of endosperm. Now all of us know that the growing embryo will completely start utilizing the endosperm. All of you know this. Growing embryo is going to completely start using up the endosperm because that is the one who is going to provide the nutrition. Now in albuminous seeds or in albuminous ones, what is going to happen? Some amount of endosperm will be retained, right? Some of the endosperm will be retained. Okay, got it all of you? Now in exalbuminous students, no endosperm will be there. Okay, X, no, past. Okay, no endosperm will be left. Okay, no endosperm will be left. That means everything will be consumed. Okay, everything will be consumed. Yes or no? Okay. Yeah, I have a cold. That's the reason. I don't know from where this climate is playing around with my life. Okay, so albuminous, there's one way to remember, Cafe Coffee Day. All of you have heard about Cafe Coffee Day, no? So Cafe Coffee Day, that is, we call it Cafe Coffee Day as CCD. Okay, CCD. So CCD. Think of this CCD. Now in this D we don't have anything, but CC we have. Okay, where do I write it, students? I'll write it on the top. Okay, no place to write it here. So example to remember is going to be Cafe Coffee Day, that is called as CCD. C for what? Castor. Another C for what, students? All of you tell me in the chats. Okay, and to remember exalbuminous. 1 second. Yes, someone is telling. What is Sudarshan asking? Doesn't even know my name, huh? Then why are you sitting here? Okay, coconut. No one told coconut. Hello, people. Okay, and for your exalbuminous, you're going to remember as, you can remember PUBG actually, but B, nothing is there. Okay, so P for P. I mean, U, nothing is there. Okay, U, nothing is there. B for bean. And G for groundnut. Okay, G for groundnut. The main problem with botany is to remember the names, examples. Okay, today the whole problem is that to remember the example. So students, you can remember it as PUBG. Okay, So when I was writing this, I was like, okay, PUBG. PUBG, that is pea, bean and groundnut. Here you can remember CCD, that is cafe coffee day, where you have castor, coconut and D for nothing. So albuminous retains some amount of endosperm, and exalbuminous no endosperm will be retained. Okay, no endosperm will be retained. Is this clear, all of you? I have also told you nuclear and cellular. Now I'll teach you, when I teach you about coconut, I'll explain you that also. Okay. Now students, all of you know we are all doing tender coconut, right? In this tender coconut, there is going to be two regions, right? There is a tender coconut, the water, tender coconut, water, okay, and then there is going to be the white kernel. That is the region that you eat. Okay, the white kernel is the portion that you eat. All of you have eaten? All of you have eaten? Yes, PUBG, huh? Okay, all of you would have eaten this. Now the tender coconut that you're drinking is the PEN. That means your nuclear, free nuclear divisions, and the white kernel that you're eating will be your cellular divisions. Got it? So the portion that you're eating is going to be your cellular endosperm, and the portion that you're drinking is going to be your nuclear endosperm. Got it? Now I don't know where to write it. Okay, so this is going to be PEN, okay, and this is going to be cellular. This is a question that has been asked very commonly about your coconut. They'll ask you about the tender coconut. Exactly, the coconut that Hasini sent, if all of you see, that is the dried one, right? Where you will see your endocarp, mesocarp and all that. But normally your tender coconut, what is the water that you drink is going to be your PEN, that is primary endosperm nuclear or your free nuclear endosperm, and the white kernel is going to be your cellular endosperm. I told you cellular endosperm is followed by a cytokinesis. That means splitting of the cytoplasm is happening. Are you all clear on this? Tell me quickly, all of you. All of you? If so, I'll proceed. Okay, if so, I'm going to proceed. Okay? Yes, very good. All of you. Okay? Yes, fine. Yes, okay. Chalo. So students, if they ask you what is the fate of the endosperm, this is the fate of the endosperm. Okay, neither there will be persistent endosperm. Okay, so fate of the endosperm is going to be where they'll have a persistent endosperm where some amount will always be retained, and then there will be complete usage of the endosperm. Okay, so fate of the endosperm is neither they become persistent or they be used up completely. So if it is persistent, write castor, coconut, rice, all of that as the example. If it is non-persistent, you can write about pea, bean and groundnut as the example. Okay, yes, okay. Fine, very good. Okay, now we are going to understand embryo. That is the next stage, right? So here you can see dicot and monocot embryo is there. This is a monocot embryo, this is the dicot embryo. So always remember, now if I draw a dicot, okay, if I draw a dicot, so this is dicot, cotyledons, two cotyledons are there. The one that is going up, okay, one that is going up and one that is going down is the most important that you have to remember. Okay, so I'll tell you that first we will try to understand monocot, then we'll come to dicot. So students, if you see the monocot, you will see a plumule region, right? A plumule region is the region that gives rise to the shoot. All of you know this? Okay, is there anyone who doesn't know this? That give rise to the shoot of the plant. Okay, then there are cotyledons. Okay, one cotyledon will be there, that is why it's monocot. Okay, then we have hypocotyle, then we have radicle. Radicle is the region that will give rise to the root. Okay, that will give rise to the root, and to protect the root there is going to be a root cap. To protect the root there is going to be a root cap. Again, this where are you going to study? You are going to study in your morphology of flowering plants. You will study, yes, you will study in morphology of flowering plants. All of you, all this you will study very, very clearly. Now, as I told you, now I have drawn one dicot, two cotyledons are there. Students, the one that is going up, okay, that will be called as epicotyle. Okay, epicotyle, and the one that is moving downwards will be called as hypocotyle. Okay, that means the one that is forming the shoot, right? Whatever is going above means that is coming above the sand, right? This is the mud, the plant is there, whatever is coming above is going to form everything about the shoot. The one that is going down is going to form whatever about the roots. So the one that is going up will be called as epicotyle, the one that is going down will be called as hypocotyle. So now if you look here, can you see hypocotyle that is moving downwards? Okay, now look at this and see the dicot. If you see the dicot, you will see the scutellum. There is actually a layer that protects or do that differentiation between your endosperm and your embryo. Okay, that differentiation will be done by your scutellum. Then you can see coleoptile. Coleoptile is the one that will protect your plumule, right? Because it's going to be forming a shoot. So coleoptile is going to be the one that are going to protect the shoot. Okay, the plumules. Okay, so coleoptile, and then you have shoot apex, epiblast, not required now, radicle, root cap. See, coleorhiza, the one that will protect the radicle is called as your coleorhiza. This detail you will study in your morphology of flowering plants. Now you just have to know that this embryo will have the fate of the embryo will be, it will form monocot and dicot. Okay, and the one that is moving upwards is called epicotyle, the one which is moving downwards is called hypocotyle. Upwards we have a plumule that will be protected by a coleoptile, then we have downwards a radicle that will be protected by a coleorhiza. Plumule will give rise to the shoot, radicle will give rise to the root. Done? So fast I told. Yes, done? Done, students? Please tell me. Yes or no? Yes or no? Done, 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 done. Yes, ma'am. Only one student. Other people, hello. What are people thinking and sitting in life? Okay, done, ma'am. Okay, so what and all did we study in embryo, students? The most important thing is, see, there is an embryogeny for your monocot and dicot. That means what is going to happen? The generative cell has fused to the egg, they are going to form the zygote. After multiple division, they are going to form embryo. Okay, now in embryo, you have to remember that there will be two types, right? There is going to be monocot and dicot embryo. Monocot and dicot embryo. Monocot and dicot embryo, they have cotyledons. Okay, cotyledons make them different. Now in your monocot embryo also, all of you remember, there's going to be one cotyledon. Okay, cotyledon. Okay, common things are going to be your plumule, that is going to give rise to your shoot. Okay, that is going to give rise to your shoot. Then they have mentioned, okay, plumule, radicle, that is going to give rise to your root. Okay, root. Then here dicot also you have two cotyledons. Okay, two cotyledons are there. And here again plumule, same thing, students. Plumule, shoot. Here they have coleoptile that will protect these people. Okay, then here you have radicle that will give rise to the root. Okay, that will give rise to the root. Here you have coleorhiza. Okay, coleorhiza. Done, done, done. Yes, epicotyle, hypocotyle. Very good, very good. Okay, cotyledons done. Plumule, epicotyle, hypocotyle. Okay, that also I am just drawing it here so that this slide is more than enough for you to study the whole thing. Okay, so this is going to be epicotyle. Students, I am writing this so that, see, when you see so many colors, you will feel like studying. Okay, if you see NCERT, you will be like, oh, who will read this now? I don't want to read. Okay, epicotyle, these are the cotyledons. Okay, cotyledons. Two. Done? Okay, ma'am, that's all. Perfect. Chalo, let's go to the next portion, that is your seed. Okay, now we did embryo, we did endosperm, now we are going to do seed and fruit. Then we are done with the chapter. Okay, done with the chapter. Okay, seed. First of all, you have to understand, students, the ovule, right, the fertilized ovule is going to form the seed. Many students will not remember this. Okay, fertilized ovule will form whom? Seed. Yes, ma'am, we agree to this. Okay, yes, ma'am, we agree to this. All of you agree to this? A fertilized ovule is a seed. Okay, so seed is what? The final product of sexual reproduction, right? In angiosperms, seed is going to be the final product of sexual reproduction in angiosperm. Okay, sexual reproduction. Yes. Now, students, seed, the main thing, first before I go into seed, I have to tell you about the seed coat. Okay, I have to tell you about the seed coat. So, if we divide the seed coat, that means to protect the seed, there is going to be seed coat, which is made up of testa and tegmen. Okay, testa and tegmen. All of you remember the ovule, no? Anatropous ovule? Yes or no? All of you remember the anatropous ovule? Ha. In that only your seed coat is going to come. Okay, in that only seed coat is going to come. So, the testa, okay, the outer one, here somewhere the arrow mark is there. Ha. This one, the outer one is going to form the testa, that is formed because of your outer integuments. Okay, this is your testa. Testa. Inside that, tegmen. Okay, tegmen. This is your, here, this one is your hilum. Okay, the small opening that is there. Fine. Again, this seed coat will come into, but students understand, testa is made because of your outer integuments. All of you remember in your ovule, you had outer integuments, inner integuments to do the protection, right? So, the testa is formed of outer integument, okay, outer integument, and this is the outer layer also, okay. Outer is testa and tegmen is made up of the inner integuments. So, now you will be able to relate to everything, otherwise you will be blank. Please remember the ovule diagram, don't get confused with the embryo sac. Okay, very good. Okay, outer integument is clear. Fine, that's all. Okay, so in the seed coat, this was the most important thing. Now, I'll label some of the parts here. Okay, I'll say label some of the parts for you people. Okay, now students, in case the ovule is unfertilized, okay, the ovule is unfertilized, okay. So, the unfertilized ovule will form an unfertilized seed, right. Fertilized ovule will form a seed, means an unfertilized ovule will form an unfertilized seed. That condition is called as apomixis. What is that condition called as? Apomixis, okay. Okay, I have one thing. So, give me 1 minute. I'll just drop a message to. Okay, 1 second, okay. Okay, yes. What in all problems, no students? Okay. Yeah, sorry. Okay, students, look here. The one that you are seeing here is going to be epicotyle, or we can say the plumule is giving rise to a shoot, okay? But I'm going to write epicotyle. Okay, epicotyle. Then here this is going to be a hypocotyle. Why are we telling hypocotyle? That means this fellow, see, you can see the leaf formation, right? The shoot is getting formed. So, this is going to be the plumule, that is going to be the radicle, okay? This whole region is going to be a cotyledons, okay? Cotyledons. Cotyledons. Yes? Done. So, this is all about your seed. Now, I told you one important thing. If an unfertilized, okay, if an unfertilized ovule, it's a very important thing, okay, if an unfertilized ovule forms an unfertilized seed, unfertilized seed, then what will it be called as? It will be called as apomixis. It will be called as apomixis. Is this clear? Is this clear? All of you in the chats? All of you in the chats, let me know. Is this clear? Okay. Now, students, why is this apomixis even being discussed? Because it's actually going to help the farmers, right? Imagine unfertilized ovule form unfertilized seed, and in angiosperm, the ultimate final product of sexual reproduction is seed. Right, the farmers need the seed. So, that time delay will be, taken into consideration. So, apomixis is actually helpful for farmers. Now, apomixis is mainly seen in examples like your orange, okay? Example orange, most of the citrus fruits you can tell, apomixis is seen. Yes. Santosh, I am teaching genetics. Okay, chalo, genetics. Yeah, I usually teach line by line. Okay, so don't take tension. So students, apomixes is actually helpful for the farmers because the ultimate product that they need is seed. So that will happen without unfertilized ovule, means then it's easy, right? So it's mainly seen in citrus fruits. Okay, I'll write in citrus. Citrus, I'll write. Citrus. Done? Done, all of you? Okay, Santosh, I can see. I read it. Okay, I read it. Students, citrus is there, mango is there, grasses is there. Multiple examples are there, but whatever you want to remember, you can remember. Now, I'll tell you about some more important points like your persistent nucellus. Okay, all of you remember the nucellus that was there in your ovule, right? Now, in case the nucellus is persistent, okay, persistent nucellus. That means nucellus was supposed to give all the content to the embryo sac, but the nucellus remained, like persistent nucellus happened. That ones you have to remember, that will be called as perisperm. Okay, that will be called as perisperm. This perisperm, so this question has been asked, whenever there is persistent nucellus, what will it be called? Perisperm. Example is going to be, you can remember B and B, okay, beet and your black pepper. Black pepper. Students, the most important thing in this chapter is that there are a lot of small examples given everywhere, so that you have to remember, like this, beet and black pepper. There it was given citrus, mango, like small examples are there, that you write and keep. Okay, at the end of the chapter when you're reading NCERT, take a sheet, for everything write an example. Now I'll teach you polyembryony, apomixis, parthenocarpy, parthenogenesis. All this you have to write an example and keep, because that time confusion will not be there. Clear? Okay, clear? So far good? Yes, very good. Black and beet. Okay, black pepper and beet is going to be there. Now same way, what is seed? So here, basically here I'm going to write all the other terms, okay? Other things I'll write later. Fruit I'll teach you now. So students, there's something called as seed dormancy. That means the seed is, like the condition is unfavorable, so the seed has just, like how do I tell, like your hibernation or your estivation, no? Seed has just, because the condition was unfavorable, the seed just stopped germinating, okay? That will be called as seed dormancy. Okay, seed dormancy. You would have studied this in your plant growth and regulators, if I believe, right? So in this, what is going to happen? The seed will not germinate. Okay, the seed will not germinate, okay, due to unfavorable condition. Due to unfavorable conditions. Like your hibernation and estivation only, where the organism sleeps when it is too cold and when it is too hot, they do not want to waste their body. Got it? Seed dormancy. Now we'll go into fruits. Okay, persistent nucellus done, seed dormancy done. Okay, let me also check your NCERT, because in this chapter now the last 2 pages are very important. Okay, sometimes we'll miss something. Okay, yes, persistent nucellus, seed dormancy done, seed done. Yes, I think everything is done. Okay, monocot, endospermic seed. Okay, yes, done students, everything is done. Right, you also please check and tell me. That's the job that you should do. Okay, seed coat done. Okay, that's all. Perfect. We have done everything. Good job to us, yes? Okay, everything is done. Okay, chalo, fruits. Fruit, students, very simple. Fertilized ovary is called fruit. Yes or no? Fertilized ovary is called fruit. Agreed? Fertilized ovary is called fruit. Okay. Now, there are, if I divide fruits, I can divide fruits into true fruits, false fruits. Okay. If I want to divide, so let me write it in this corner. Fruits, I'll divide it into parthenocarpic also. True fruits, okay, false fruits and parthenocarpic fruits. Parthenocarpic fruit. Now ma'am, what is this false fruit, true fruits, like your true ribs, false ribs? Students, true fruit means fertilized ovary should form fruit. If fertilized ovary is not forming fruit, I will obviously call it false fruit. Agreed or not? Agreed or not? Polyembryony, next slide I'll teach you, okay? Okay, look here. True fruits, false fruit, parthenocarpic fruits. True fruits means they develop from the ovary itself. Okay, ovary will form the fruit. False fruit means except ovary. Okay, except ovary, it is formed from any other part of the plant. Like example, thalamus. Okay, example, thalamus. Thalamus forms the apple. Okay, what we eat, no, is the thalamus of the flower. We're not eating the ovary. So example, apple, strawberry. Okay. That means anything except the ovary. Actually, this became too much color, students. 1 second. I think I'll write a little above, but I can't move this also. Everything is a problem. Okay, wait. I'm writing it once again here. Too much yellow. Yes, thalamus. Perfect. Very good. Okay, thalamus. Okay, I'm writing it again here. Ovary. Okay, ovary will form the fruit. Okay, got it? False fruit means anything except the ovary. Okay, ovary is not forming it except the ovary, like your thalamus. Examples. Okay, examples is your apple, strawberry. Here you can write mango. True fruits, you can write mango, kiwi. Okay, these are all true fruits. Here it is strawberry, apple. Done? All of you are able to see? Yes, all of you are putting the images. So nice of you people. Okay. Okay. Now what is this parthenocarpic fruit? Students, without fertilization, okay, without fertilization, these people will be forming the fruit. Okay, without fertilization, they will form the fruit. Example is your banana. Example is your banana. This parthenocarpy also is very helpful for the farmers because without fertilization, they will be able to form the fruit, right? So example is banana. So this is very important, parthenocarpy. Okay, parthenocarpy, false fruit, true fruits. All of you are with me on this? Okay? Yes. So students, here what is happening? We can induce this with hormones also. Okay, we can induce this with hormones. So this is where no fertilization happens but still yields us the fruit. That is what the ultimate is, no? That's what the ultimate is. So that here except ovary, other parts are forming like a strawberry, apple where thalamus and all will be formed. Here ovary forms the fruit. Exact true fruits, example mango and kiwi. Mango and kiwi. Okay, done? Done and dusted? Any doubts? Any doubts? Ask me now. Now one more small portion, okay? Very small portion. Again, this is study in morphology. Some teachers actually skip this here. But since I am not the botany teacher, I will not skip it here. Again, Baswa sir will teach it again for you, okay? So here, what is going to happen? If we divide the fruit, no, we can divide them into 3 parts will be there. Fruits. All of you know this for sure. Morphology is not done, no, students. We have to do, no? Okay. Epicarp, mesocarp, endocarp. This all of you know, right? Mesocarp, epicarp, mesocarp and endocarp. All of you? Yes. Okay, epicarp will be the most outer one, mesocarp will be the middle one, endocarp will be the most inner one. Okay. Now again, fleshy, fruity, that depends on the fruit to fruit. Okay, fruit to fruit. Now coconut it is different, mango it is different. Okay. Okay. Now, if you see epicarp, it's actually the wall of the ovary. Then your mesocarp is the fleshy part. Even the endocarp could be fleshy in some, but mostly stony in most of them. Okay, you would have learnt your drupe fruit and all that. Okay, so here, students, this is the wall of the ovary. Wall of the ovary. This will be the fleshy part that we actually consume. Okay, and this will be the stony part where the seed will be there, right? The stony part in the mango, all of you would have thought, no? So wall of the ovary, fleshy part, stony part, but fruits to fruits, little variation will be there. Coconut and mango you would have seen in your morphology, drupe fruit they call. That you will learn it later. Okay, done? Now last topic is, students, polyembryony. Last topic is polyembryony. Okay, I want all of you to check your NCERT once again. Students, NCERT is God. If NCERT is not there, then we are all, He's the God of us. Okay, so all of you check NCERT and tell me. I am also checking NCERT. Done. Students, if I am referring NCERT, you should also be referring NCERT, okay? Seed viability, apomixis. Yeah, apomixis is done. Okay, only polyembryony is left. Okay? Yeah. So, students, here what is happening? In each ovule, many embryos will be present. Okay? So, one ovule will have many embryo. Excuse me. Okay, one ovule will have many embryo. That is because the integuments would have developed, the nucellus will have developed, all this happens. Okay? Yes or no? Okay, so here one ovule will have many embryo. That is polyembryony. Clear? This will be mainly seen in your citrus. Okay? So this is because, why this is happening? Because integuments can develop. Okay? Integuments can develop, nucellus can develop. From all these people, embryos will be formed. Integuments, nucellus. Okay? All these people will develop, because of which multiple embryos will be there. Example is your citrus. Okay? Citrus fruits. Citrus fruit. NCERT has given mango. Mango also they have given, students. Just check and tell me. Okay? Mango. Citrus fruits and mango. Students, look here. This is your polyembryony. Can you see? Multiple embryos. Okay? Multiple embryos. See, look here. This is your integuments, right? Integuments. Okay? This is your nucellus. Okay? So here you can see many embryos are there. Okay? This is the zygote embryo, the main embryo. Okay? This is the zygote embryo. This is the endosperm. This is the endosperm. The green color that you're seeing is the endosperm. Got it? And then you can see here nucellus embryo. Okay? That means embryo which are formed other than the zygote embryo, other embryos. Is this clear? Only citrus fruit is given, no? Okay, chalo. Students, so polyembryony means where there is multiple embryos in one ovule. This is because integuments, nucellus all take part in formation of the embryo. That is the reason, and this is seen in citrus fruit as well as in some varieties of mango. I hope this was clear. Let me check. Students, everything I have written, okay? Every single thing I have written, and also put NCERT snips. So there is nothing more, okay? There is nothing more. The whole chapter is done.